So now we shall be discussing PCR. So before PCR, let's look at replication itself. PCR is essentially a replication process. So so let's look at what all we discussed during replication. Replication means we had discussed how helicase would unravel it. 1 second.
Bro, I'll just be going on mute for a minute. Okay, some disturbance is going to come up, but I'm listening. Just continue.
Yeah, okay. So we had talked about how helicase will separate. In replication, helicase would uncoil it, and you would have DNA polymerase, which would start coding here, and then it would keep on coding till the end. ठीक है. So that was how replication actually happens, and yeah. So you have DNA polymerase, which is doing the polymerization, and we have DNA ligase, which is ligating the broken bonds. Now, remember both of these like enzymes, because we are going to use them in biotechnology. DNA-dependent DNA polymerase and DNA ligase. Just remember what they do. Ligates, that is joins. DNA-dependent DNA polymerase produces DNA. Cool. Hello. Yeah. So that is basically the recap of the process that you require before you start with PCR. So PCR refers to polymerase in reaction. So polymerase should give it away, right? Polymerase हमने कहाँ सुना था? DNA polymerase. Chain reaction, like something that is exponentially growing is considered to be a chain reaction, right? So remember we had talked about hydrogen bonding in DNA, that on heating it will sort of separate out as different strands. So you have like DNA, apply heat, then you have individual strands, remove heat, you have now during our DNA polymerization, first step was unraveling, but instead of unraveling we can just apply heat, the entire coil will be unraveled, and then we can sort of do a replication here. So instead of just removing heat directly, we do a side step, that is we allow this thing to be replicated by a polymerase, so that new strands are being produced. Actually, this will be sort of separated out. It is separated out, right? Actually, also.
Right. The exact same DNA replication that you explained earlier.
DNA replication, and so on, and eventually it will make. तुमने हीट प्रोवाइड किया, ये सेपरेट हुआ, ठीक है. एनवायरनमेंट में डीएनए पॉलीमरेज था, तो डीएनए पॉलीमरेज स्टार्टेड रेप्लिकेटिंग इट. सो इवेंचुअली यू विल एंड अप हैविंग, एंड साइमल्टेनियसली तुम टेम्परेचर भी कम कर देते हो, ठीक है. यू ऑल्सो रिमूव टेम्परेचर, तो तुम्हारा कुछ ऐसा बनेगा. सो 1 से 2 हुआ, 2 से 4 बनेगा, 4 से 8 बनेगा. सो इट्स बेसिकली 2 raised to a power, right? It's multiplying by 2 every time. It's exponential of 2. So one more thing, like यहाँ पर when we first discussed replication, we had 2 strands, correct? First question you would ask is that replication has 2 strands. Here there are no 2 strands. Wouldn't that affect the functionality? These are far away, right? As long as DNA polymerase comes, right, there's only one strand. So won't this cause issues?
Oh, okay. It needs that origin site, right?
It needs that origin site. And origin site is looked at both things, right, together. Like in
Right.
in RNA, it was only one string. So it needs that initiation sort of sequence to happen.
So
Right.
wait, do you introduce this and then put heat so that the origin site is fixed and then it's separate?
Kind of. So what we do is that we
Yeah, both ones. That is.
so what we do is that we add small sequences called primers. Primers are basically small DNA sequences which are like corresponding to complementing some of the DNA of the region of interest. And then we do polymerase. Yes.
Interesting.
Like you add primers in such a way that it's complemented, and then DNA polymerase feels like there are already two things. So let me just continue making stuff.
I see. Huh. Bro, but for this,
Cool.
you need to
So
know the sequence all of the DNA
itself,
before
you need
starting
to know
this, right?
sequence. Yes, you need to know what sequence it is to decide upon the primer.
Okay.
Okay, you cannot arbitrarily take a primer and expect it to work. Yes, this is a pre-existing knowledge. So you have something called primer banks, which are basically like given the DNA string, they will sort of make the primers for you and so on.
Mm-hmm.
Because and this requires knowledge. So PCR requires knowledge. So let me recap the entire process once again. You have DNA.
Cloning, right?
You want
DNA
to
cloning.
multiply, amplify. You want to amplify DNA. So amplification is the main purpose. Uh-huh. RT-PCR is real-time polymerase chain reaction. Nothing special about it. RT stands for real-time.
So wait, I think I have some logic in mind. Just tell me if this is correct. Okay.
RT-PCR.
So,
So what is happening is that
huh.
you do a PCR, you amplify it, and PCR can be used for. There is a way of using PCR to sequence, which we haven't come to yet. So currently, I'm just discussing amplification. Starting with PCR, right? This process of amplification is the most general use of PCR. Okay, so that's why I was rewriting the entire equation so that I can explain the basic process in the new head. You have your original DNA strand. You heat it up. It becomes two strands. There will be some primer which is corresponding to it. So primers will sort of attach, and it will be here. And there will be polymerase is somewhere here. Next thing you know, like this segment is also being built. So it will look something like cool. Hello.
Yeah,
Yeah,
got it.
and then you remove.
And you remove the heat.
Yeah, and you essentially get two DNA strands.
Right.
Fine. So what I can actually do is that suppose I have this long DNA, and I'm not interested in entirety of it. I just want a small segment to be repeated, to be sort of coded for. What I do is that I choose my primers very carefully. I start my primer from here.
Yeah, okay.
Start my primer from here. So everything that will be expressed will be from here to whatever is ahead, right?
Mm-hmm.
So for a like double-stranded DNA, double-stranded. So for a double-stranded DNA, corresponding my chain DNA, I can, if I flank my fibers, suppose this is my region of interest, this small box is my region of interest. I'm interested in genes only here. I can intelligently choose primers such that my primers are not at the end. Okay, let me make it sufficiently long. Like imagine it's very long. Because only then it makes sense, right?
Right.
I have a
Right.
small region that I'm interested. So I can intelligently choose some primer which starts here, like this. So that only something between this region will be replicated.
So, but it will replicate from the starting point till that end of the strand, right? You're still, I mean, your small
So,
part is part of it, but won't it go till the end?
it won't. So the reason is that so actually this has to do with the property of uh, the way polymerase works also. Like when these are separated out, like these are not happening in isolation. Like क्या कहते हैं, your primer. So in the replication fork, like actually this is a question that I should have covered in replication fork itself. Remember I said that replication forks look something like this.
Right.
ये वाला चीज ऊपर जा रहा है. So question is नीचे क्या हो रहा है? Correct. That is a very valid question, right? नीचे like नीचे actually some other place there will be another replication fork.
Right. You can have multiple sites.
Yeah. So what will happen is that it will replicate till somewhere it will stop. So the primer selection is so that your stop is already within the gene of interest. Like there's a stop place, right? There are two ORI sites. You select the ORI site closest to your gene of interest. It might not be this close. Like मैंने ये case दिखाया कितना close है, but actually it may not be this close.
Okay.
So your coding is actually like your replication is actually happening from one ORI site to the other ORI site.
Okay. Okay. So it's ORI site to ORI site, but not exactly in that place. Your like your region of interest should lie between two ORI sites. That's it.
Two ORI sites, yes. So now I don't have anything here which is corresponding to यहाँ का sequence, right? My polymerase, there is no polymerase attaching here. It will basically make an DNA till here, and nothing will be made here.
No, but what if you encounter an ORI site in the region of interest? Would that be an issue?
So that is why you have the control of primer, right?
Yeah. No. What I'm saying is,
You can have multiple primers.
within the red box that you drew, what if there exists an ORI site in that red box?
So you expect it to terminate in between, right?
Yeah.
Doesn't matter. You have primers. You can add another primer.
Oh, okay. That's what you mean.
So, Shez, like imagine a DNA.
So wait, I think I understood and I know how to explain it to him. Okay. Like see, you see that the DNA polymerase, right? It opens it up only to a small region. It doesn't open up the entire DNA, right? So the region that it opens up is from one particular ORI site to the other ORI site because one DNA polymerase opens up this region and the other one is working on the other ORI site in parallel. So what's happening is if one site is opened up and then like you're not applying heat specifically in this point. Okay, imagine that situation. So the other site is also being opened up and worked in parallel. So there, in like you can imagine it as the in whatever place those two converge, right? These two sites, that part is still connected. Assume it that way. So what is happening is now that you have your region of interest should lie between two ORI sites, and there can be several other ones in between them. In case there are several others in between them, you start adding more primers so that you do that part in parallel.
Correct. So
Then it'll terminate, right?
terminate. It will make smaller sequence. Arey bro, boy, that's why I made this image. So imagine you are working in some kind of like Shreyas as a sweeper. Like Arihan को warehouse worker बनाया था, Shreyas को sweeper बनाएंगे. ठीक है? You are working with your colleagues in this thing. Like you have people stationed here. This is you, okay? You have decided कि कौन सा region कौन करेगा. You just have to go from here, your section, to the section where the next person will start from. Correct. तुम उसके section में भी झाड़ू नहीं लगाओगे. ठीक है, within this platform. You will keep on sweeping this section. And you will, इस point पे आने के बाद there might be a pole or something, and you're like, oh, this is next person's site, so I won't sweep here. मेरा काम है नहीं. Why should I do it? First thing. हाँ. Terminates. Now, how do you imagine primer? अब poles थे ना between each sections, right? Imagine that you need to like तुम काम तभी करोगे जब तुम्हारा पैसे दिए wages are put. ठीक है? Why would you work for free? Right? So एक दिन support generally सबको सबके पोल के वहाँ पर पैसे रखे होते हैं. So everyone gets their wages. Someday, one day you come out and you see तुम्हारे like थोड़े लोगों के अंदर है, थोड़े लोगों के नहीं है. तो क्या करेंगे? जिनके पास पैसे वो झाड़ू लगाएंगे अपने एरिया में and they'll go. That is exactly what is happening. That is the role of primer. झाड़ू लगाना is corresponding to your making of DNA. अगर तुम्हें कंटिन्यूसली सपोज तुम्हें इन दो सेक्शन में करना है, लाइक इन दो सेक्शन में झाड़ू लगाना, so you will put a payment thing here. Correct. So then you will have contiguous section of where it is swept. This is corresponding to the contiguous section of DNA that is being produced. Similar thing you will do in the opposite also. Does it make sense?
Yeah.
This is where there is a reason. A prime, 3 prime. Sorry, 3 prime.
Yeah,
3 prime.
it's
5 prime. Look at our Ori wala diagram, bro. It was moving in one direction, right? This stand is missing. Cool. We don't want the staggered production at all. पर स्टैंड नहीं है तो स्टैगर्ड प्रोडक्शन हो ही नहीं रहा. So now you understand, right? No, they are not connected to each other. These are two independent things. This Ori side is connected to this termination side actually. Actually, I should have made something like this. This Ori side of the second strand is same as the termination Ori side of the first strand. Does it make sense? इन दोनों का एक साथ होना चाहिए था, but your non-staggered वाला sequence goes only like this, right? This यहाँ पर stagger करके नहीं जाएगा वो. Non-staggered इस तरीके से जाएगा ना. Production is happening in this direction.
Wait, that is correct. But wait 1 second. So in this diagram, these two are actually very far apart, right? These two strands during this year.
Yeah.
Yeah. So
Yeah.
isn't the original diagram actually correct? Like before you change the colors of the primer, because whatever you see from 3 prime to 5 prime on the first one would be the 3 prime on this side should actually be. Yeah, you just have to flip. So the original coloring was
I think the color was confusing. Like, that's it. This region of interest, like, I can just sort of flip this entire thing, right? Arihant is
Yeah,
completely
yeah,
right.
correct. I mean, just because you change the color, I was like little bit concerned on
या
what
5
happened.
प्राइम टू 3 प्राइम है. अगर मैं उल्टा कर दूं, डीएनए में उल्टा सीधा कुछ नहीं होता ना. The
Yeah, it's
directionality
the
is
exact
matters. ये
same
भी 3
thing.
प्राइम टू 5 प्राइम है.
No, no, I'll tell you. Like he changed it from purple to green, right? So I assume it's like a different thing you have to work with because it's a termination site, but it's technically the same thing but flipped up.
Same as well. Yeah. So the reason why I explained it specifically is that I said that it has to be flanked by it. Right. So this is what I meant by flanking it. So इस वाले में कैसा स्ट्रिंग बनेगा तुम्हारा? One DNA will be made from this side to this end, right? इस वाले के लिए this side to this end. मतलब ये बनेगा. और ये वाला पार्ट नहीं होगा. You will end up having blue DNA. Like actually you will end up having this long DNA. अरे
I mean, I got only that particular sequence
with
of blue.
blue
Yeah,
DNA.
only that
Yeah.
short blue sequence.
But next time around, because this blue is also separating it out, so you will have this is for one cycle, right? Long sequence and blue. But so first round, this is what it will look like. Color blues thing. Next time around, you will have something like
Everything will separate out, right? You'll have more of the blue stuff going.
yes.
And you'll have
But
a blue
like
blue pair.
इसके साथ भी separation हो.
Yeah, blue pair.
Two blue pairs, right?
Right. Yeah, the ones you.
Yeah. Okay. Oh, do you want the other blue then? One second.
No, we got it. No, we got
It
it.
will look like I am using blue for the shorter thing, right? That's why I use the same colors.
Yeah, we got it.
I'm just showing
The shorter blue
shorter,
was nice. Yeah, shorter blue was nice. That was fine.
Yeah, so this is short.
And then
you
have
end up having
two short sequences.
So you're running it for multiple cycles, right? And this is exponentiation.
Right, so you'll see
and
a lot of
no
those
matter
short
how much you
blues.
exponentiate, your wastage is only 2 strands. If you're looking in scales of thousands, hundreds, 2 is already
Right.
negligible. Thousands, it's negligible. But in PCR, we end up getting billions.
Right,
So
if you run
imagine
it
how
with like
negligible
30
it is.
cycles, you get more than a billion pairs
Yeah.
of those short ones.
Exactly. So 30 by the time 30 cycles is done, this is so negligible that you can ignore it completely. This strand do not exist. Essentially, you're
Right.
getting blue strands. It's the region of like your sequence of interest. Bro, नीचे वाला ओरी साइड ऊपर वाला ओरी साइड नहीं होता. There is ओरी साइड. That's it.
Yeah, it's just an ORI site. So I got your confusion, bro, because see, I had the same thing. But then don't even look at the complement stand, okay? Because that is literally the same stand, but in the opposite direction. That's what you think of it as. So like this is the complement stand for now. But no, I'm saying don't think about the complement stand right now because
There
that
is
is
one ORI site
confusing to
here.
think about.
There is another ORI site here. Correct. Cool. What you are forgetting is that ORI site is not in one string. It is on both the strings. And ORI site is not like your AUG. It is slightly more complicated. That's why I'm not getting into the nitty-gritty details of it. Just assume that it can identify the origins of replication.
Okay, so the ORI site for one particular strand, and even though the reverse strand has its complement and stuff, that part would still be recognized as an ORI site, right? At the same location.
Yeah.
Is
So
that the
it
thing?
recognizes this as a ORI site. It doesn't recognize this as a termination site or something. Okay, देख गया कि ORI site है, so it terminates. Actually, I think this thing was also causing an issue.
No, I think I understood Shreyas's doubt. His thing was that will the ORI side be the same even if you take the reverse complement on the other stand? Like at the same location. I think that's what he meant.
So, like, it's not on the same location, right? पूरा मतलब
Oh, okay.
both are the same. Like Ori is Ori, like
Okay.
Ori and the Blind Forest. Yeah. I don't know. Like we have been discussing this for hours, and it didn't strike me till now. So this sweeper example kind of explains it, right?
Yeah.
Imagine each individual strand as a sweeper wala case, like with primer being the money. ठीक है.
Oh, that's really
Now,
smart, bro. Like you're leaving the exponentiation to do its job.
exactly. It's you see how amazing this is, right? Like because you're literally doing exponentiation with DNA, and you're just exploiting the fact that it's making twice, two copies, again and again and again.
Exactly. It's not like you're putting in a lot of work. Your work is minimal. You just have to start this process and it'll just do its work.
And that is why it's called chain reaction, right? Once you start a chain reaction, it just blows up.
Yeah.
Literally, what's happening
Nice.
here. So, I mean, two things we have already mentioned what should be there in this mixture. One is our DNA of interest, of course, it should be there. Next is our polymerase, should be there. What is our primers actually, because there will be multiple type of primers. But like for simplicity, let's consider there is a primer. Like that generally doesn't happen. There are multiple primers. So primers, okay, yeah, primers. So you have your DNA, polymerase, primer. What else do you need within the mixture? What do you think you need?
Some kind of heat controlling stuff.
Heat control is
Oh,
right. I'm
okay. You
talking about
mean
chemical
in
material.
terms of DNA? Okay.
Huh.
No, look, I remember that question from the second assignment of about some TAC polymerase and DDD and TP, some stuff, but
Yeah,
I don't know
Taq
exactly.
polymerase I'll come to. Taq polymerase is same as our polymerase. Why it is
Oh,
Taq
okay.
polymerase, I'll explain. Come to that. What else do we need? कुछ नहीं चाहिए.
Yeah, that's
So
about it, right?
अगर मैं इन
Oh
तीनों
wait,
को
do we
पानी
need
में
a
मिक्स करूँगा,
ligase?
ligase is also not required. No, ligase is not required, right? Because fragment is not being made. Fragments are not being made. Bro, but चेन बनने के लिए न्यूक्लियोटाइड्स कहाँ से आएंगे? कौन बना रहा है न्यूक्लियोटाइड? पॉलीमरेज में
Oh
न्यूक्लियोटाइड
no!
कहाँ से मिलेगा ब्रो? Bro, but
No, no, it
it
does
has to
not.
be in the environment, right? It should
Yeah,
be there in your first
so
step, right?
until now it was there in the cytoplasm or some place where it was picking it up.
Nucleoplasm. It was there within your
Nucleoplasm.
nucleus
Yeah.
ka, plasma. So that you have to add nucleotides. DNA hai, so you'll have DNTPs, which is basically your A, T, G, C, which will blending blocks. Actually, I should put this in blue because we are using blue notation, right? A, T, G, C, which will be used to make the new DNA. ठीक है? So these are stuff that you need to put in your
I see.
polymerase chain reaction. Now let's come to polymerase. Like as I said, the heat is sufficiently high to break down DNA. Now polymerase is a what? It is a protein. It's an enzyme, right? Enzymatic protein. So do you think where DNA is getting broken down, protein will not get affected? It should, right? Naturally, you expect it to
Yeah.
get denatured or something. Yeah. So like actually in most of the cases polymerase just get broken up. So what we do is that we use a specialized type of polymerase called Taq polymerase.
This
So
is what I've
Taq
seen in
polymerase
this.
comes from Thermus aquaticus. So Thermus aquaticus is it's a bacteria which is in hot water spring. Bacteria in hot water springs.
Oh.
So it has a special type of polymerase which can withstand high temperatures.
I see.
Nature has every other ingredient that we needed for this technology. Think of how cool that is. We just brought together different stuff and boom, we are doing it on our own. And this sort of adds, like last time ITBM, I went into origin of life and all, right? Like abiogenesis. So like this is also something that points towards abiogenesis. We are able to do biological reactions on test tubes on our own using chemicals which okay have some link to life, but like we are making it on our own. How is it? Without any biological organism per se in it, with just chemicals, we are making new biological compounds. So Thermus aquaticus bacteria's polymerase is used, and Thermus aquaticus, that's how Taq comes from, TAQ. Like Thermus aquaticus polymerase. Yeah. So I think अभी 2 बज रहा है. PCR तो कवर हो जाएगा. Like Sanger and tracing will also be covered. Like sequencing, PCR based sequencing will be done.
So cool. Then we'll do one thing. Like we'll do it until 2:30, and then since all of us have OS, right? We'll all read like one part of OS and like explain it to each other tomorrow. Because I guess all of us are doing OS tomorrow, right?
Bro,
And then
I think it like once this much is covered, like अभी जल्दी sequencing cover हो जाएगा. I think we can cover the entire thing also. Like let's try. Like might take till 45, but cover हो जाएगा.
Good.
I don't think it's a like there's too much to cover now. Actually, there is. So like I'll cover till PCR and sequencing, and then maybe we'll take it up. So Taq polymerase comes from Thermus aquaticus bacterium, which has polymerase which is resistant to your what do you call your heat. So that's it. So you have DNTPs. DNTPs are what? Your ATGCs basically with sugar. So these are building blocks for your new RNA. Fine. Now we come to sequencing techniques. Like what is the purpose of amplification? Amplification is because we had very few amount of DNA. We need to generate enough DNA so that we can actually send it through some machinery and recognize it. So the common machineries are basically ion trap based methods. That is like you ionize like different like ATGC in different manners, and you you check at what speeds they're dropping down in terms of sequences. And
right, this is a gel electrophoresis thing, right?
No. Like we haven't read gel electrophoresis, or you do radiation method. Gel electrophoresis is closer to the radiation method and Sanger sequencing. One basic way of sequencing is that I make this radioactive TGCs that I'm adding. The DNTPs here are radioactive. I have what radioactive nitrogen. That is the easiest thing I can do, right? Radioactive nitrogen.
You mean
radioactive
fluorescent, right? Like so that they show different colors.
Fluorescence is light, right?
Yeah, is it radioactive or is it fluorescent?
It's fluorescent. It's fluorescent. My bad.
Right. Because I
Ionization
remember
method
reading
is
fluorescent.
ionization. Yeah, it is fluorescent. Bro, my brain is also not good. Fluorescent.
Fluorescence.
Yeah, so you add fluorescence to each of ATGCs, and what you do is that once you have sufficient in numbers, you basically cut it into you basically shine light upon it, and you sort of get the sequence. But the thing is that your DNA sequence is not straight, right? It's coiled up and wound up and whatnot. So how will you directly count like this? The basic sequencing method is really inefficient and it doesn't count properly. Like you can't directly sequence it. So normal PCR is practically of no use when you're looking at sequencing. Instead, you have something known as Sanger sequencing, which is the most popular PCR-based sequencing technique.
Great. So, PCR was useful only to amplify and create several copies, but it's not useful for reading the sequence.
Not directly. Like you have to make some modification to PCR technique to make it useful for sequencing.
Okay.
That technique is called Sanger sequencing. Sanger sequencing works like this. You have the same starting products. You have this normal, non-fluorescent ATGC, and you add fluorescent DD and TPS. Okay, so what are these DD and TPS? Yeah, DD and TPS stands for dideoxy nucleotides. Now, what is so special about दोनों OS निकल गया? It looks something like this. Does it make sense? What does it mean? Like, अगर DNTP, like, if your polymerase by mistake places the DNTP instead of NTP, your chain will terminate abruptly.
Right.
The polymerase is putting DNTPs one एक एक करके, right? It's saying A, T, G, C, A, T, A करके कुछ कर रहा होगा, and अचानक उसके पास GNTP आएगा, G. Now it cannot continue, right? Because there is nothing to continue.
Right.
No OH group continue, so it abruptly stops. इससे क्या होगा? You get short fragments, every possible this thing, containing every possible lengths of it. What do I mean by this? Suppose the sequence that you want to replicate was A, T, G, A, T, A, T, G, C, or A, T, C, G, something of this order. ठीक है. This DNTPs can come in anywhere, right? So you will have some amounts of just A sequence. Yes.
Wait, what? I didn't get it.
Oh.
Okay. You're saying put one at every single spot so that you'll have all possible substring. Oh, so you're saying it's probable that you'll have every single substring.
You will have, right? There are NTPs in the solution, and तुम बार-बार कर रहे हो. Notice that NTPs, DD NTPs are the only ones which are made ionic. So I think fluorescence has something to do with radiation. Like it is made fluorescent through some radiation technique. I'm not sure of it, but it is like light is shined on it. That is what I know. That is how it's said. What is happening is that only thing which reacts on light being shined on it are the ones with our GD NTPs, right? Correct. DDNTPs, DD,
Ah.
DDNTPs. Sorry, like I said GD by mistake. DDNTPs. My throat is also good. DDNTPs, one by one. Like, see, every stand will have only one DDNTP, which is the terminating
Right.
point. And these are the fluorescent ones. Keep this in mind. नहीं हो सकता ना, because it's starting from your primer, right? Did you forget about Ori site and all? Oh. And we fixed it. So no other combination is possible except for smaller fragments with terminating sequences are DD and TP,
No, but
which are
you're
made
kind of
fluorescent.
you're kind of relying totally based on probability of which one is picking, right? Either DNTP or DDNTP. But it's just that there are so many,
So
like you can have billions of them, such that all of these ones would form at some point.
yeah, they will, right? If you have billions of things, even with if you have 50% of all,
Hmm.
and you expect at least 50% of DDNT, like not 50, you will end up having exponential 50, so you'll have around 12% of this thing, right? 50 into 50 yoga of
No, the thing
DDNTPs.
is, see, right now you have all these with you, but if your sequence
Correct.
is really, really wrong, sorry, long, right? From like the sequence that you got from your PCR itself is very long. So
ठीक है.
while picking these things, it can so happen that some of the ones might not even occur, right? Like one of these substrings might not occur at all.
Because you run out of material or something. Is that what you mean?
No, not only material, but even if you take a billion possibilities, maybe one out of 10 tests. Yeah, it's like that chance is there that you might not get it right. Like
Bro,
that
this is all
possibility
of
is
these
still there.
techniques are error prone. These are prone to error. Like it doesn't give you perfect results every time.
Ah, okay, okay. Yeah, that was the thing. So it's like,
Yeah.
yeah, it's prone to error.
Everything is so. Yeah, assignment me. You would have read something like PCR is prone to probabilistic errors because
Hmm.
probabilistically it can
That to NGS something.
and
I know, or
no
CDN,
no
something
PCR
related to
to
that.
cloning technique. PCR to cloning technique and cDNA to NGS. This was the questions. PCR and cloning, bro, vector cloning. PCR, these both are amplification techniques. Cloning means something completely different in biology sense, right? This is called amplification, not cloning. Like, not this, ये जो sequencing में पहुंच गया, but what we are doing earlier is called amplification. हाँ. अब, so you are getting, this is more prone, right, to errors because it's probabilistic.
Hmm.
Like, there is, each thing, it's like a coin flip with very high
Right, right.
tendency of going correct, but it's like still a coin flip.
Correct.
Uh,
Priya, so you're saying that based on the last DDNTP, that is the one that's actually fluorescent. So when you shine light on it, that is what
no,
indicates, right?
you don't directly shine light, right? Now, this is all happening in one test tube.
Hmm.
तुम टेस्ट ट्यूब पे शाइन लाइट करोगे तो कुछ आएगा ही नहीं. Now, all of these DDNTPs have different colors, obviously. Otherwise, we won't have. There's
Hmm.
no purpose of right. So I should have used different colors, I suppose.
But like all A's would be same color, right? Or those will also
Yeah.
be different.
No, no. All A's will have same color.
Okay. Yeah, yeah, but that was understood. Yeah.
Oh, okay. Wrong color. G C F. something like this. Cool. All A's are of the same color, all T's are of the same color, fluorescent color, all G's are of the same color. Cool. I'm not recalling thing, bro, but for some reason I believe that this was also done by some like fluorescence. कैसे add हुआ था? It had something to do with radiation.
Yeah.
It is fluorescent. Arihant is correct, but like I'm getting confused because of that.
Bro, I remember
Arihant is correct.
fluorescence because I read it before this assignment only. That's why I
Like
remember it's fluorescent.
it is fluorescent. Like I'm getting confused why I remember it as radiation.
Ah, okay.
Like that's all. I mean, memory is a fickle friend. Maybe I'm overthinking this, but yeah.
Huh.
Fluorescence is correct. Yeah. So what you do is that this is all lumped together. You can't separate it out so beautifully like I have done, right? ये सब कुछ ऐसे tangled mess है.
Yeah.
Okay. So what do? You have to separate it
Oh,
out.
this was the part where the like longer the string, the slower it flows or something.
Yes, yes. DNA has what charge?
Uh, I don't know. Phosphate has can go up to +3 if you lose something. Oh, right. PO3 is wasn't PO3 minus.
Negative charge.
Yeah,
Yeah.
negative. Huh. PO3 is
Yeah.
minus some -3
3
of.
minus. Yeah. It has a negative charge. What we do is that it's a process called gel electrophoresis.
Ah, right. This was the part.
What is done is that you have wells here, like starting wells, and you put your sample using some pipette or something in each of these wells. Actually, for the timing, just consider that this is one huge well because I think that would be easier to explain. Imagine this is one huge well and you're putting a sample here. Take a sample. No, you're forgetting that DNA is like each base has one negative charge. So that is why DNA coils around this thing, right? Your histone protein. Histone octamer is 8 positive charge. So you put it in wells. This is also known as southern blotting technique, by the way. Not important. So what you do is that you put it in this well. ठीक है. And you apply charge. That is, you apply negative charge here and positive charge here. Now DNA is negative. It gets repelled to negative charge here, forced to move forward, and it's attracted to positive charge here. Now the shorter strings have less mass, so they are very free to come here. While the longer string will take more time to reach.
Right.
In essence, what you will have is that you will have these band patterns.
But isn't this the same thing we saw with that eco array cutting thing in that REB site? Sequence stuff itself. Like basically, it was basically cutting on restriction enzyme sites. Right? Yeah, yeah, yeah.
So you realize what is happening here. Your entire sequence like this is being obtained.
Yeah.
Yeah.
You'll get like every single length, right? So you'll get one
The
color
exact sequence.
after the other. Yeah, if you just look at these colors, you can get the entire sequence.
This is called Sanger sequence.
We had Sanger sequencing in ITP, right?
Yeah.
Yeah,
So
same.
I taught you then also. You don't remember. but इतनी डिटेल में नहीं किया था because इतना important नहीं था. He just mentioned Sanger sequencing. So
No, see,
I,
that time we didn't
I
do.
remember doing gel electrophoresis with you before.
Gel electrophoresis you taught. We didn't teach, like we didn't have Sanger sequencing. That's the reason I remember gel electrophoresis. I don't even remember the word Sanger sequencing.
Like all of this is very smart. Like nothing. Like you are letting DNA do its job in a very smart way, and you're utilizing those properties to
Yeah.
do stuff. So this is called Sanger sequencing, and this was how Human Genome Project के लिए भी sequencing and all was done. And she mentioned something known as Celera sequencing, which is based on blasting it. Like instead of having DNA like this, like instead of doing a PCR and then doing a Sanger, you just blast it into pieces. And then like if it's a, and then you try to sequence. So you will get, instead of you agitate, basically तुम्हारा ये होगा, you vibrate it a lot at very high frequencies, then your bonds start breaking, right? It's a chemical, right?
Hmm.
At a certain resonance point, your bonds will start breaking.
But wait, how did that help though?
It doesn't, like, it just, because Sanger ka issue was that it is noisy, that is one. For longer strings, as Arihant pointed out. And second thing is that it's also expensive. You have to put in long gel electrophoresis strips, and you need to do all of that stuff, right? And you cannot parallelly process multiple parts. So what you can do is that you can run it on these separate fragments. To broken
Oh, okay.
fragments.
And then use some computational method to stitch it together.
Yeah. So remember, ma'am was talking about whether we did jigsaw puzzles, and like will jigsaw puzzles be better if we have smaller, more, less. So this is only done for shorter sequences. That is the disadvantage of Celera sequencing.
Yeah, I mean,
Like,
clearly, if you look at it, I think you just have to, you'll get all these pieces together and you find all possible, what do you say,
combinations.
combinations.
Yeah, and
Oh, I'm sure.
whichever one seems most perfect, like you continue with it. So
Yeah, so
that is Celera sequencing. Next generation sequencing. Break करने के बाद you do Sanger. You don't directly do full sequencing. कम हो जाएगा, एंड कॉस्ट भी तो कम हो रहा है तुम्हारा. यू हैव टू पुट इन लेस डीएनटीपीस. डीएनटीपीस को फ्लोरेसेंट बनाना था ना. दैट वाज द बिग चैलेंज. दैट वाज वन.
wait,
एंड सेकंड,
1 second. In this process, right? Like you were starting with A and then going on, and then you just have all of them together. But in this case, you had only one. You're taking one sequence. You broke it into pieces. You already have the pieces, right? Where did the DDNT piece come in? Like when does it attach
bro,
to
we
it?
have these pieces, but we can't. How will we sequence it? Read the pieces, right? How will you read the pieces?
Yeah, see, I got that you have pieces, and those pieces would be put into gel electrophoresis, right? So you will know the exact sequence of the entire. Oh, okay, you're saying do the exact same
No,
thing
no,
but with
no.
smaller pieces.
But with smaller pieces.
Oh, okay.
Also, your
वॉशरूम जाके आ। अरे जीशान
also तुम्हारा polymerase का write process जो था, was longer, right? Because वो linearly कर रहा है. Like it writes each base in linear time. Correct, if that makes sense. एक एक करके हर एक बेस को लिख रहा है ना. मतलब if you have a string, it is
Yeah, yeah.
reading it linearly. So if you have 10
Correct.
length string, it will take 10 times as much for it to do 100 length.
Mm.
But here, because you have smaller fragments, like parallel processing.
Kind of doing parallelly.
You're doing it parallelly. You don't. So that is
That's the
the
problem.
disadvantage. That is the problem. You just try out all the combinations. Easy. This is cheaper, right? It's like parallel processing. मतलब तुम्हारा polymerase will take less time. Instead of while it goes through billions of bases, instead of going through billions of bases, a billion bases together, it will go 1,000 bases simultaneously. How billion is 10
So I
power
think
6, right?
in some way, in some sense, computational power is cheaper, right?
Less time, right? Think of it like instead of going to 10 to the power 6, order of 10 to the power 6, we have loaded to order of 10 to the power 3. So it's actually log n sort of thing. Linear OC. Celera. So Celera people have this advantage कि human genome project has already happened. लोगों ने Sanger sequencing करके ढूंढ लिया कि क्या क्या genes possible है. So they know what they're looking for. The search problem is minimized. Yeah. They just have to make sure which part fits. Like उन्हें बस jigsaw puzzles वाला काम करना है बस. That's it. So that is Sanger sequencing, Celera sequencing. Now we come to the OPS methods of all because it is mind blowing and it's very, very high level. So even I can't explain. Like, alag level, high level. It's called NGS techniques.
Interesting.
Yeah. Next generation sequencing methods. Next generation sequencing methods is a very broad term. It refers to lots of different things. So I won't be going into details of it because no one cares like कि क्या details है because it's too complicated. Like it's very cutting edge and very complicated. So one method is ion trap. Second is pyro sequencing. And third, I don't remember. Pyro sequencing, ion trap. I think यहाँ पर radiation वाला था. I'm sure.
You should teach any of the club.
No, I don't think so. So that is why I said I won't go into detail. This is way beyond any of what we can do because नहीं NGS mention किया था ना like so that's why I'm covering it. Like I'm not going into each of the techniques because of it. To put it very simply, what you're doing in ion this thing is that each of these have a specific charge, right? Each of these bases, like they behave differently to. Wow. Okay, my handwriting is very bad. Sorry.
It's fine.
I'm not sure of. One second, let me search NGSK techniques. Trap, I remember because NGS is.
So yeah, the specific methods is not an issue. That is fine.
Yeah. See, massively parallel signature, polynomial pyro sequencing, ion torrent. Yeah, ion torrent is the ion trap method. Like I think so. But this has nothing to do with the other ion trap. Yeah. So like there are lots of methods and like that are done for considered part of NGS. NGS is NGS is OP, next level OP stuff that is happening. Iron trap I'm going to explain because I like iron trap a lot. Iron torrent method it's also called. So basically it has two blocks which are ionizing blocks. Like there's a electric gradient in the downward direction and there's a sort of trap here. And there is a binder ion stream that is going like this. So basically what happens is that your DNA string is forced to pass through this in a straight manner. This ion beam makes it stop long enough so that the reader can read from here and recognize what thing it is. It's like
Did you explain this to me before?
I'm
or
not
did
sure.
she explain? Or I think
She might have
maybe
explained,
for yeah.
bro.
I don't
She
remember.
has. It's is there in the lecture slides?
Huh.
This is Oxford nanopore wala sequencing, which is OP level, and that is what
Right.
that what is the reader for my. Like I'm connecting this to my research because my research may be exactly same.
Because I remember seeing this diagram or hearing this explanation because I went through her slides.
Yeah, she might have 1 second. Yeah, it is there. It's there.
There, right? Yeah. Seems familiar. I think that is the complicated part.
That is exactly the wrong sort of question because I won't be able to answer. Yeah.
No, but what was the?
So you see this, right? DNA helix is force feeded into this ion trap, and like there's an adapter which sort of reads it. There's an ion beam which goes like this, which prevents it from going. See, flow of ions through the pores. So ion beam blocks it and keeps it long enough for the adapter molecule to bind and read. So, and this is how small it is. Like actually, this doesn't. Yeah. And this doesn't do justice to how small it actually is. So, minion. So, like I was there in the lecture and I felt that it's not doing justice, so I sent it on C&D group also.
Yeah, you sent this.
It's laptop and that's it. How OP is technology, bro? It is pretty expensive.
Yeah, that is crazy.
It is pronounced as minion, bro. Min ion ke jagah, it is pronounced as minion. So it's basically Oxford nanopore technology. Oxford is the one who they are the group who made this technology. So this NGS is a large term. It involves lots of high-end technologies that I don't think we'll ever use. Like even she can't cover because it's way beyond our patterns. So the advantages of NGS is basically this, that you can do RNA, like cDNA technique only allows mRNA. Like
yeah, I have no clue what
mRNA.
cDNA is.
So what cDNA technique is that you have RNA, then it will convert to complement DNA, and then do Sanger sequencing on the DNA. Because the reason is that mRNA are long enough. How it could happen is that तुम्हारे पास एक सैंपल है एंड सैंपल के अंदर तुम्हारे पास प्रोटीन्स होंगे या sugars होंगे, fats होंगे, all of this you have to digest, right? So you put stuff that you put proteases, you put sugar consuming stuff, put fat consuming stuff, so that you have only RNA left or RNA and DNA left. DNA also you will delete using specific sort of enzymes. DNA नहीं है actually, that's why you are using RNA sort of thing. But now what your sample remains has small tRNA sequences. tRNA and rRNA are much small in comparison to mRNA. So you are unable to sort of convert it. Like you are not unable to read and make complementary DNA at all in this process because these are too small to detect. Which is long enough. Yeah. So, like, if you want me to use technical terms, the unprocessed mRNA is the true mRNA, like before introns are cut. After introns are cut, it's called hnRNA.
I see.
So, like, heterogeneous, like non-nuclear RNA. So, sorry,
No,
this
but
is
wait.
unprocessed RNA.
Uh,
This is unprocessed RNA, and this is mRNA. My bad. hnRNA. Heterogeneous nuclear RNA. Nuclear
but
is
wait.
understandable, it's happening in nucleus.
No, but see, one thing that we did until now is you can use DNA to create more DNA. You can use DNA to generate RNA, and whatever generated RNA is there, it codes for amino acids, and those make protein. But nowhere did you see RNA giving back DNA, right? Like, how is that process even done?
Correct. So let's go back to origin of life itself. How do you think DNA came in the first place? I told you early life was all RNA, right?
Right.
You're all double
Uh,
stranded RNA, single stranded RNA protein.
hmm.
Correct. How did we go to DNA in the first place?
Oh wait, was it that RNA was unstable as a single strand, so it gets oxidized
See,
and you
that got
get
selected because of natural selection. Like that happened because of
uh,
natural selection. मतलब mutations बहुत हो रहे थे, so DNA-based organisms started surviving more. But how did it actually
hmm.
happen? We ended up making enzyme complexes that reverse this. Enzymes exist which do RNA to DNA. So
Oh,
actually, you can guess the name also. नाम क्या है?
RNA dependent DNA polymer or some RNA dependent something polymer. Yeah.
That's it. But the issue is here is that RNA-dependent DNA polymerase is like cannot really work with short sequences. It doesn't really
Yeah.
matter कि mRNA की कौन सा RNA. As long as it's long enough, it can work with it. If it's short, it can't. As simple as that.
Yeah, yeah, we understand.
Okay. Technique. So you have RNA, you convert to DNA, and then you do Sanger. That is cDNA technique. So what you
So
do in
Sanger
NGS
was
is that
basically whatever sequence you have, you like you without breaking it into parts, you just try to find all possible stuff by attaching DDNTPs and then do gel electrophoresis, right? That
substance.
is Sanger.
Yeah. Yes,
Okay,
correct.
huh. Consider all substance and then gel electrophoresis.
So where was I? Huh. So cDNA हुआ. Uh, अब like what NGS does is that it's like okay, I don't need to even convert RNA back to DNA. Few of the technique directly sequence your RNA.
Yeah,
Like because just think of how Oxford was working also, right? It's
yeah,
reading the
just
DNA.
yeah,
It didn't do any sort of stuff on the DNA, right? It forced it through a membrane and it's just reading it. Similar thing is happening here. And उसमें फर्क नहीं पड़ता कि छोटा है बड़ा है, doesn't matter. You can read everything. हाँ. So that is one advantage NGS has, and second is it has it is something what is known as high throughput. Sequencing, which basically means that your resolution is high as one nucleotide. Like you can distinguish between two nucleotides that are very close together. Like, even if you have one random floating nucleotide, even that can be read. Or if you have two nucleotides thing, you can separate between कौन सा first था, कौन सा second था.
Okay.
So the resolution is very high. So NGS is like top of the line sort of thing. Like the actual science behind it, मैं नहीं explain कर सकता because I'll require a PhD for just explaining it.
That's fine.
So like I from what I can see, she has like written about NGS applications. So it includes. Oh, did I mention NGS is cheaper also? And Sanger. फ्लोरेसेंस करना था, जेल इलेक्ट्रोफोरेसिस करना टाइम लगता है एंड यस या सो हाउ OP is that like cost has reduced like anything and it's become faster and in every sense it's become better. Yes, of course. So anyway, so NGS के applications लिखा है like epigenomics, that is basically methylation patterns. Remember, this was my topic of ITB term paper. Like your genome का thing is not just dependent upon the DNA itself. It's also based on where methyl groups are attaching. So NGS, some NGS techniques can detect where methyl groups are attaching also. Not only DNA, it can also tell कि methylation कहाँ हो रहा है. It can also tell कि histones में क्या differences and so on. So NGS is very high-end sort of technique.
Wait, I forgot what methylation is. If you methylate
Methylation
DNA, what happens?
No. Methylation is what like I did my ITV project on. So it turns out that you have DNA sequence. Earlier the understanding was DNA sequence. As you have this promoter site or initiator site, promoter, right, for DNA. So you will have corresponding RNA till terminator, and this RNA will code for some protein. As easy as that. So wherever you see a promoter, there will be an RNA corresponding it, a protein corresponding to it. Then it turned out it doesn't work like that. Certain areas in the DNA are surrounded by CH3 group, which is methyl groups.
Okay.
And presence of these methyl group prevents those proteins from being expressed.
Oh, I see. So that part won't be a part of the RNA.
Yeah. And this methylation is directly dependent on what kind of environment you're in. Like, is it too hot? Is it too cold? Are you under stress? Are you under... It is.
Yes, I am under stress.
So, like, lots of work has been done with twins, like, or cloned, like, cells and, or cloned organisms for that matter. Methylation. Turns out different conditions have different effects. So, like, even if
This
your
is the
genetic
single
data,
most coolest thing.
even your genetics doesn't describe who you are. Your environment and genetics together have to work together to make you.
Interesting.
So even if you're like like you can't create the same individual without replicating the entire
Environment.
thing. Yeah. Second, HTS NGS, like you would see that she's done it in alternative ways, like alternate करके slides में लिखा हुआ. But it's actually the same thing. HTS is same as NGS, high throughput sequencing.
This lady really needs to get her slides also right. When I read this, I didn't understand the word.
Like I can see where people can get confused. यहाँ पर HTS लिखा है, then NGS. HTS.
Yeah.
NGS and HGS are the same. Like
Okay.
NGS and HGS are actually very large terms, like in the sense that one term catches all sort of thing. Like there are lots of technologies in it.
High throughput.
High throughput sequencing. That is high throughput. I explained, right? High throughput means that you have resolutions up to 1 nucleotide. Yeah. NGS also means this. I'm sorry.
I think we're done.
Bus vector cloning.
Right.
Only
vector
cloning, cloning and VNTRs are left. So like I'll just complete with NGS and that's it. So vector cloning and VNTR, like Wales, all the things she was trying to explain. Yes. Bro, I've been speaking for how many hours continuously now?
Yeah. Less than 1.5 hour almost. Yeah. Yeah, bro. I'm down, bro. I've learned so much today that I haven't learned in the entire semester until now.
Like you can freshen up after finishing. Fine, like that way there's less pressure on you also, right? फिर मैं like बस OS में focus करेगा. Like
Yeah, good.
so like let me just finish with NTS. Like this is one more application and then we'll finish. So next application is metagenomic sequencing, which is basically you compare sequences between different species.
Okay.
So basically, how do you know that monkeys and we correlate, or what is the relation between us and a banana, or stuff like that? Yeah. So what you do is that because NGS makes everything fast and cheap, you can basically build an entire library of sequences, every single organism that's there. So and you can compare across species also, right? So
Something like 90% match or something, right? Similarity.
and even more than that, like you can have for example, imagine stuff like lichens. You know lichens, right? Lichens are what?
I'll tell you. I know what this is, but I cannot
It's
recall.
fungus
There
and
is
algae.
water body.
They are
Right.
found near water bodies.
Water body.
Fungus plus algae.
That's a
It's a
symbiotic
symbiotic
relationship.
relationship. So it's fungus and algae का symbiotic relationship. So what if you want? Do you know that lichen can that the algae and fungus also exchange genetic materials?
I did not know that.
Similarly, bacteria colonies exchange genetic material, even if it's different bacteria altogether. That's why, like, I'm going into vector cloning or like small part. Remember, they said plasmid DNA, like bacteria later. Hello.
Right, plasmid DNA. What?
So you have the circular DNA, right, where you're inserting your strands. The circular DNA is called plasmid. Purpose of plasmid is for antibody, like antibiotic resistance and other environmental based resistance. Okay. Anyway, this is an application, so I want to cover it. So what I'll do is that I'll cover stress take light and freshen up, and like then I'll start with vector cloning. Anyway, okay. The point is that you can do sequencing across like fungus and algae का exchange हो रहा है. So what you do is that you don't do sequencing of algae separate, fungus separate. You do it together. Similarly, अगर तुम्हारा stomach है, gut के अंदर you will have microbes, right? There will be different types of microbes also. But they are exchanging genetic information either and other. So you don't sequence them separately because then you'll end up losing out information. You sequence them together. Yeah, you treat them as one organism. You treat them. It's not actually one organism. You just treat them like one organism. So all of this falls under metagenomics. Kind of. I mean, I think she just. No, she mentioned metagenomics, and metagenomics is not a very straightforward thing, so that's why I mentioned it. Otherwise, like, if you know me, right, I like more of the theoretical stuff here. Like, even biotech wala stuff, I'm not very interested in. Like, I'm very interested in जो शुरू में हम लोग कर रहे थे कि कैसे ये हो रहा है. Like, I'm interested in the fundamentals of biology rather than what is actually happening in tech.
Yeah, looking at it and getting my mind blown. That part is amazing.
Like, Alapan, like, for example, you have a DNA sequence, say, ATGCATA, whatever,
Yes.
ठीक है. हाँ, इसके corresponding तुम्हारा होगा, this is from
RNA की
5 prime.
AUG A
नहीं, RNA नहीं, मैं इसका corresponding DNA पहले लिखूँगा, TA,
अच्छा उल्टा
T, TA, T,
C
C,
D
G.
T A.
Correct. 3 prime to 5 prime. Now, what will be the? Now, my question is this: like, why do you think? अच्छा, एक DNA स्ट्रैंड से कितने RNA बनता है? Like, एक डबल स्ट्रैंडेड DNA स्ट्रैंड से कितने RNA बनेंगे at a time?
स्टैंडर्ड DNA से कितने RNA बन सकते हैं?
एक ही
Feels
बनता है.
like should be two, but yeah, क्यों एक क्यों?
It should be two, right? Like, it makes more sense कि दो अलग information है, तो दो अलग RNA के लिए कोड करना चाहिए. Correct. But actually नहीं होता. Have you ever thought why नहीं होता?
नहीं.
So the reason is this, like, तुम्हारे पास suppose होता तो क्या होता? Like, this thing will code for an RNA that will code for a protein that you want,
Yeah.
but the opposite will code for a protein that you don't need. So it's a wasteful process. That is one reason. Like, we don't want to code for RNA that we don't require. Yeah.
Yeah,
So
but
the
that is
second
not
one
a
is not-
problem.
Yeah, that is not a proper answer. Second
Yeah,
answer
that is just explaining evolution kind of. Yeah, like I don't.
yeah. I mean, that is also a thing, right? It's a wasteful process, so it makes sense why you won't do it, right? Evolution doesn't want you to waste your resources.
Yeah, but like it just doesn't feel correct.
Yeah.
Like it feels like you
Now
know
let's
everything,
consider.
now you are deriving the solution.
Now let's assume that दोनों से RNA बन रहा होता. ठीक है. So what will be the RNA sequence?
AUG.
It will be
Sorry, USC D B C G U A S. अरे T U. यूरेसिल डाल दो.
oh sorry, my bad. Sorry, like I was copying this one, so इस चक्कर में होगा. इसका क्या होगा?
उल्टा जस्ट
नीचे वाले का
ऊपर वाले का उल्टा. AUG
A G. And this is from 3 prime to 5 prime, right?
Yeah.
And this will be from 5 prime to 3 prime.
Yes.
C U A
Right.
C G A U.
So I think something messed up.
हाँ
CG
ओके सॉरी
बीच में CG GC होएगा.
इसलिए मेरा सीजी इतना खराब आ रहा है जीसी और सीजी को मिक्सअप करो
Wait, how the fuck did
हाँ
that?
So in between one C, like AUG, and then it shouldn't be C. It should
नहीं,
be A again.
ये उल्टा है. हाँ, G होएगा ही नहीं. U
AUGA.
AUG इधर G. हाँ,
A.
yes
हाँ.
नहीं, yes wait नहीं.
Yeah, this
हाँ
is
हाँ
fine.
हाँ yes हाँ हाँ. Yes.
This is fine.
now it's fine I am dumb bro like I don't know my brain is also not working क्या हो रहा है हाँ अगर ये होता do you see the issue already do you see the issue already
नहीं,
Yeah.
एक दूसरे का उल्टा है मतलब redundant है.
Redundant,
redundant नहीं है
yes.
ना
This is what I thought in the
bro
beginning, but
it's
then I
not
had my mind
redundant
blown.
bro no, it's not redundant नहीं है it's a completely different thing
Yeah.
या AUG का corresponding एक होगा.
But now if you look at it,
But 5 prime से but नहीं but coding 5 prime से नहीं चालू होता. कोडिंग 5 प्राइम से नहीं चालू होगा.
हाँ
तो अगर 5 प्राइम
yes
से कोडिंग चालू होगा
प्राइम से
तो
चालू हुआ
दोनों का. अरे फक, ये तो पहले उल्टा कॉमन नहीं है. अरे शिट.
common नहीं है bro
हाँ हाँ
it's not
अरे
a
नहीं नहीं.
palindromic sequence
हाँ हाँ
just
अरे
seems so because AUG से
हाँ
start हो रहा है
हाँ
like I
अरे
realize
फक.
that it's a bad example
हाँ
it's not
हाँ
the same sequence I got when I flipped it I realized that ऐसा लग रहा है कि
या
palindromic
हाँ
है
हाँ
okay let's screw this thing up a bit so this will become
हाँ ये completely different from Linux.
GC so ये G
बट ये डबल हेलिक्स होता क्यों है? जस्ट टू हैव अ स्टेबलर वर्जन.
oh because there is like attraction also right stabler version is why it is advantageous to us why it happens is chemistry pure chemistry because those strands have electronegative groups they are just attaching with hydrogen bonds
बट लाइक व्हाई इज इट कि इक्वल नंबर ऑफ ऑपोजिट पेयर्स है ही.
equal number of opposite pairs मतलब
लाइक so like one is useful, another
I
is
don't think so
useless,
like
but the other one loves it, so it's just keep it. And it's also good because
oh you mean oh, why do we have it in the first place itself right
yeah
bro that is for stability
हाँ cool हाँ फिर सही
like
है. हाँ
हाँ,
like
evolutionary
मतलब what.
purpose में this is adding stability double strandedness directly adds
अब
stability
it makes sense. It's like having a
now while we are on the topic of double strandedness अभी तक तुमने notice नहीं किया this is going to form a double strand
yeah. Now it's coming up.
yeah वो दोनों attach कर लेंगे.
हाँ so we will have a double stranded RNA and double stranded RNA is useless because अब तुम्हें इसे uncoil करना पड़ेगा and protein को करना पड़ेगा but uncoiling like I went into details of why uncoiling is different I spent quite some time and hence the effect was much higher at that point but like uncoiling is a very energetic process you can't just randomly uncoil
But
RNA
then
like this.
how do you only get convert RNA of this one DNA and not convert the other one?
Because only one DNA has like promoted initiated or promoted स्टार्ट करेगा.
तो लिटरली
हाँ.
तो एक बार ही एग्जिस्ट करता है और ओनली एक ही स्टैंड पे एग्जिस्ट करता है.
एक बार नहीं, मल्टीपल बार एग्जिस्ट कर
So you
सकता
can
है.
have it in
अलग अलग जगह
multiple
पे
points.
बट लाइक
Example-
दूसरे स्टैंड में एग्जिस्ट करता ही नहीं है.
पता ही नहीं है.
What the fuck? This is
हाँ.
doesn't this feel
Like-
like someone coded it? Like जैसे
Yes, God
It's an
coded it.
else if loop, bro. Like if I need this protein, bind here. If I need
No,
the other
it's
protein,
like-
bind here.
it's like how OS starts, right? How तेरा boot वाला program finds OS का location और वहां से start करना पड़ेगा.
Like I there's lots of computer science connection within biology. Like biology gives lots of ideas about computation. Like even look at our replication fork wala thing.
No, but that is
I mean-
more of application, right? But
So
only from the theory
bioinformatics-
aspect,
Yeah, I'm
you can
talking
learn a
about
lot.
not- मैं उल्टा तरीके से बोल रहा हूँ. Like bioinformatics is application of informatics for bio, right? You're using informatics in bio. I'm talking about biology for computer science.
It makes sense, right? Like computer science is kind of studying formal computational systems, and like organism
You have a
is
brilliant
like
computational system right in front of you.
yeah, and an organism is like the best possible computation system.
This is going to be a systems biology course. The idea that computers are all systems. Yeah.
कितना बाकी है तुम
Yeah.
लोग का?
Only vector clone. Yeah, okay. And
Actually,
vector cloning.
vector cloning mainly. Vector cloning will take slightly more time. मतलब you
Yeah.
because it contains stuff like restriction enzymes and all also. So
No restriction enzymes light. I read it today, and then the restriction mapping also I learned how to do it. There's one really good YouTube video on that.
Restriction enzyme is
Yeah.
really easy. Like हम लोग एक दिन बैठ के पढ़. Yes, because I want to do
so
something.
like to reach vector cloning wala thing, first we have to realize how bacteria are. Like bacteria live in generally very harsh environments.
Yes.
Like there will be different kinds of chemicals.
When they're scared, they
There'll be
form
other
the
kind. So there'll be different kinds of chemicals. There'll be different kinds of what do you call thermal issues like heat ज़्यादा होगा कम होगा.
Are those
Or-
cilia?
I just wanted to make the bacteria look beautiful. Bacteria don't have like cilia, right? They'll have cilia. Bacteria will never have cilia because cilia are eukaryotes on the outside.
Bacteria to prokaryote. Wait, some bacteria
So,
like no bacteria are. All bacteria are.
Bro, all
Algae-
bacteria are prokaryotes. In fact, bacteria have gone as extreme as like being considered too archaic to even be considered prokaryotes. Like there's a concept of archibacteria, right?
Yeah.
Archibacteria. That is that they are older than prokaryotes. They are this that entire collection was made just to make separate them out as extremely old creatures.
Yeah. Wait, mitochondria is like
It's
archibacteria.
mitochondria is supposed to be like adaptation of archibacteria. Like
Huh.
that is, it's a very controversial topic in biology, but yeah, that is
But
what
it
lots
makes
of people
sense,
believe.
right? It has its own DNA.
Yeah, but it need not be archibacteria, right? It can be some other bacteria which sort of got into autobiotic relationship.
Yeah, that is also possible.
Yeah, so chemical thermal, there will be biological factors also. Like for example, there might be viruses in the environment or fungi. So antibiotics that we eat, आते कहाँ से हैं? They are sourced from fungi, right?
Yeah.
Yeah,
You know about this, right? How penicillin was discovered.
amoxicillin and penicillin. Yeah.
Yeah. So how penicillin was discovered is that basically Alexander Fleming was doing this thing, like he was culturing bacterium in uh, in a petri dish, and
Yeah.
like generally everyone keeps it in very high hygiene and all, but Alexander Fleming had a very disappointing day, so he didn't clean the culture, and like culture is made of this sweet sucrose agar gel thing, which is basically it's a sweet gel. So like if you put anything sweet outside, something will get attached there. So next day when he came there was mold growing on it. But he noticed that because mold were growing on it, like around the mold there is a patch where bacterium was not growing. And he correlated both of these. And he realized that the fungus must be releasing some substance which is causing bacteria to die. And that's how penicillin was discovered. It is the first antibiotic drug. So using chemical compounds in another organism to fight some other organism. So, so why I'm mentioning is that you have natural antibiotics all around. So antibiotics is not another thing it has to be afraid of. ठीक है. So our bacterium which lives in such a hostile environment, what does it do? Like, so how it happened? Like, there are so many types of hostility, right? How does it brace itself for different hostility? It has to survive also, right? So it
Bro,
does
this
it.
is scary. I am just going on noobs. Okay. I don't know. I get a panic when I read about this.
Okay, bro, it is a very happy story. So stay around. It's a story of teamwork, please.
How do you guys read about these things and not feel like these can attack you and you can die anymore?
Bro, I mean, it's there everywhere
Dying won't be too
in your
bad, right?
surrounding.
Don't you want to live forever? Serious.
Yeah.
So just think of this, like you have large bacterial colonies, okay? That is,
Yes.
bacteria are always in colony. You won't see one in isolation like anything.
Yeah.
But
If they're in
bacteria,
isolation, they become sad and create.
they have less chances of survival. That is why they don't go into
Yes.
isolation.
Together they're strong.
Why they are
I am
strong?
the official
The
leader.
reason is this thing called plasmid. Bacteria have two kinds of DNA. One is their main genome, which is in a nucleoid region, and they have a separate type of DNA called plasmid.
Yeah.
It's a circular thing. Plasmid, plasmid is explicitly for fighting these environmental factors. So what happens is that, like, of course, there is not one plasmid. There are multiple circular plasmids in one bacteria. So what happens is that suppose one environmental factor comes in place, say antibiotic aa gaya. Huh? So initially cells will die, but one of them would have this plasmid, right? That will immediately recognize that this antibiotic is in place. It will start replicating those plasmids and releasing it in the environment. So it will send out plasmids, and the bacteria around it will take it in, like bahar wala plasmid ko andar le lega, because it also has to survive. So if you keep a plasmid outside near a bacteria, it more or less will keep it, put it inside. It's like a survival mechanism for it.
Okay.
You're getting what I'm saying, right?
But what is a plasmid? It's still genetic material.
It's nothing but a circular DNA.
Yeah. इसका भी amazing story है. You will do cloning and stuff with this.
Yes.
You will cut it at exact position.
Yeah,
And
this
create
entire
all-
story of plasmid gives us a unique opportunity, right? Like we
Yeah.
know that antibiotic के presence में ये plasmid को ये करते रहता. Second thing, we know that the bacterium will take any plasmid around it, right?
Yes.
So if we isolate the plasmid, add some,
So उसको अब अपन बैक्टीरिया को अब अपन फेक इंफॉर्मेशन फीड करेंगे and it will become our.
How this actually works?
You are a sex deprived kid.
No.
You,
Arju, can I please explain? Like you know the answers, but like वो थोड़ा build up करना पड़ेगा so that
yes.
it's
Yes.
like understandable.
So wait,
So,
antibiotic
the way-
is anything that will harm the bacteria, right?
It's not. It's a
So-
chemical that will harm the bacteria. Like
Yeah, so when it recognizes threat, it will just take any plasmid that it finds as a defense mechanism.
no. So what it does is that it already has plasmids. So I'll come back to why they are in colonies. So those who didn't have this factor, plasmids can be thought of as stuff like sort of like कहते हैं. How do I make a good analogy for this?
नहीं, नहीं,
Kind of,
Yeah.
but थोड़ा नहीं.
नहीं, जैसे तेरा
पूरे
red
colony
blood
में
cells में कोई DNA नहीं होता. Plasmid is like pure DNA.
रेड ब्लड सेल्स इम्यून सिस्टम में काम भी नहीं करते.
Hmm?
Red blood cells don't work in immune system, right?
Yeah, white blood cells.
Yeah. Anyway, so what I was saying is this. तुम्हारा एंटीबायोटिक मैंने सपोज दिस इज योर कल्चर, ठीक है, कल्चर मैंने सपोज एंटीबायोटिक कोई कॉर्नर से इंट्रोड्यूस किया और इसके ऊपर इसके अंदर डाल दिया. सो व्हाट विल हैपन इज दैट इमीडिएटली लाइक bacterium will start to die because it finds the presence of antibiotic. But because the bacterium colony is in thousands and millions and so on, so there will be some bacteria or the other which will have the plasmid to survive this. As soon as that recognizes that this antibiotic is there, वो क्या करता है, इसको बार बार बनने लग जाता है. It starts replicating this plasmid again and again and releasing it in the environment. Why is it releasing it in the environment and replicating and releasing it? वो तो survive कर रहा है, but he is trying to save the rest of the species.
Oh,
Isn't it cool?
It's a question of species survival here, right there. So it's like this. Suppose कल मानो like अचानक तुम्हारे पास like you are getting attacked by enemies, ठीक है. So like thankfully around your house you had a fort. Yeah. So you are safe from enemies. So what you will do? You will build up resources to make another fort, so that more people will be saved, and then again and again and again. Something similar is happening. But you are sending plasmids so that the species as a whole can be done. So bacteria, as part of its defense mechanism, will take anything, any plasmid which is floating around in its medium, because it
I
thinks
have
that
a
it's
question. Why does the plasmid, like, how does the plasmid provide like antibiotic resistance?
yeah, that would have been my last question after everything.
plasmid provide antibiotic resistance because what are essentially antibiotics? There will be some kind of proteins, correct, which are doing some kind of protein structures, right? Like protein activity, right? It is either breaking down bacteria cell wall. Generally, cell wall attack is what is generally happening.
Yes.
So it will start producing counter proteins, which will either consume these, sort of break these proteins, or it can be any kind of thing, bro. Like it will either change material of cell wall in certain way so that it is.
Holy shit! So the one जिसका plasmid is good enough to protect against this kind of protein वाला antibiotic जो मेरे सामने है, that will produce more of those plasmids because it is surviving, and and बाकी चीजें भी वही plasmid लेगा and will keep on reproducing that more, so that ultimately now they will just.
Yes.
Nice.
That is insane.
So like this is exactly how like if you study your immune system, this is actually very similar to immune system. That's why I agreed with Shreyas when he said it's immune system. So immune system में क्या होता है कि you have redundant like this thing lymphocytes which don't do anything at all. Like in fact there's a Karkazad video which released very recently, and it talks about this that you have lots of lymphocytes which do nothing, which are basically just there. These are called helper T cells. So what happens is that helper T cells will have sort of every machinery to combat any kind of threat your body can ever face. Like it might sound like a bold claim, I'm saying any kind of threat, but it's actually that it can do anything. So what happens is that whenever you get a new infection, your helper T cell corresponding to that infection gets activated. And once it gets activated, it becomes a memory cell, and it gives that information to other helper T cells, and it educates other like lymphocytes and macrophages on how to deal with the pathogen.
I see.
So, like something similar is happening. Like this plasmid might have been just useless for the bacterium, but yes. So it's not protein; it's just a circular DNA.
Right. I think that should be.
So when I'm making circular DNA, like it doesn't actually look like this. DNA is always a helix, so it will look something like. You get my point, right? Like something of this form.
Yeah, joined end to end.
Yes, joined end to end. That is why it's called circular. It's like a ring, literally a ring.
Okay.
So that is our story of plasmid. Now we like as a defense mechanism, it will take any plasmid in its surrounding because it thinks that it is something that will help me in survival. So remember I said that I will put toxins in this corner, right? Why it's important that you will find that those who are around here who have never touched the toxins will all have plasmids because something would have released it. So like it preemptively protects itself in a way. However, like it can also happen that enough is not spread and then antibiotic sort of overwhelms. And that's how you actually kill bacteria, right? Bacterial infections. You eat antibiotics. And this is why they tell you not to skip doses in antibiotics. Because
okay, so if
numbers will not go down.
Start culturing again.
They'll start sending plasmids. They'll start gaining resistance. Exactly. So you don't want that to happen.
Oh, no.
This is where this concept of super germs also comes.
I mean, this is exactly why I was forever scared of bacteria. Yeah,
but like it turns out there is a cool way of beating. I'm going off topic here. Like there's a cool way of beating antibiotic resistance. That is introduce another kind of threat. Like so, what new scientists are doing is that they're adding bacteriophages.
Yeah, what are bacteriophages? Is it to like distract the bacteria from spreading a different plasmid?
No, it's literally like a virus जो तेरे bacteria को गोली मार के उड़ा दे. Boom.
So what it does is that it starts making plasmid for the bacteriophage. But because it has to decide, like it cannot express both of them equally, there's a crunch of resources. They either get killed by the antibiotic or the bacteriophage. So,
No.
Like, no, you're introducing another species that is also a threat to this, so it has to create two different types of plasmids.
Yeah, but then it has to pick amount of resources.
Yeah, but it has to pick one of them, so it might end up dying by the other.
Yeah, it's like
it is actually made for antibiotic resistance. Matlab, it is showing resistance, so you introduce this. So, like, actually, in most cases, redundant plasmids are sort of pushed away, and very few redundants are kept. But like redundants are kept. That is why this entire idea of plasmid comes from. So, like, lots of interesting studies happen in this, like, in the sense that in this sort of plasmic sort of exchange, like, it's also considered a way of sexual reproduction also, right? Because it's going from one individual to other, like literally protein is transferring. So, like, it's also very interesting in terms of that sense. And you will see that higher organisms like yeast, for example, yeast also have similar mechanism, except they don't directly use plasmids. They have another sort of vector. There, it's sort of like reproduction sort of thing. Like, they join together and exchange factors, that is genetic material. Yes. Bro, think of this. If the bacteria is in like in a it's in a peaceful environment, okay, what will it want to do? It will eat up and it will try to split into more pieces.
Mm.
But if the bacteria is in a hostile environment, it won't split. It will try to survive.
Mm.
So splitting, like, that is reproduction, is when survival is sort of ensured. It will never
Right. It's in a
reproduce.
comfortable environment.
Yeah, it's only in the comfort of resources.
One day will
Yeah.
come when we can defeat literally any bacteria, and like they aren't that scary. They are scary if you have immunodeficiency diseases,
Yeah.
which
So,
requires genetic problems, which I probably don't have. So yeah.
so are you sure you don't want me to teach restriction enzymes?
No, no, it's
Are you
fine.
not
They
stressed?
just give a. No, I didn't say I knew it. I just knew restriction mapping because I went through that video today.
ऋषि
Okay,
ऋषि
so
वही ये ऐसे बोलना वो जो हाँ
हाँ
yes yes
नहीं मैंने मैंने नहीं बताया अभी. I haven't told reached there yet. अभी मैं background fix कर रहा हूँ. So here you have a cell which A is willing to take any genetic material that you put next to it. That is one. Correct. Which is able to replicate that genetic material again and again. Okay. Cool. So what you do is that you uh, like just remember this for the time being. Okay. And so you basically utilize this property. You try to insert our genetic material within this plasmid so that you can feed it to a bacterium and make it spread. So that is what vector cloning is.
bro imagine that ये plasmid is the dick of the bacteria.
You understood like vector cloning का concept.
Yeah, yeah. I understood the concept. We'll keep replicating it and sending more copies.
So, so generally, how your bacterium looks like is that, like plasmid will look like is that there will be an oocyte. Okay, I should have used razor. Oocyte. You know what oocyte is now, so I don't need to explain that.
Mm.
We'll have multiple places where restriction enzymes can cut. So BAN H1 or EcoR1 करके होगा. There will be a section which codes for antibiotic resistance. Yeah, and there will be more restriction enzyme sites. So I think bioinserting the whole cake, I should show the actual image. One second. why is this coming back? Why did I- Did I close it or something? No, you know that. Yeah. So this is how you do it. Like you have a vector, you insert it, and then you add it. So how this? I'm trying to show how this would look like. I think it comes on later. Yeah. You see this, right?
Hmm.
This is equalized cloning vector. So it has your ORI site, ORI, and like uh, amp R is ampicillin resistance, tet R is like tetracillin, I think, ampicillin and tetracillin resistance, and you have cut sites here and here. So you can use the cut sites to insert whatever your gene of interest is. Ampicillin resistance and tetracillin resistance sites are there. So in essence, you have three, like sort of three things of interest. That is, one is your ORI site. Why is ORI site important? Because you need replication to happen. Second is your restriction enzyme recognition sites. Third is your antibiotic sites, resistance sites. Sites. Cool. These are the three things you have. Now, this is how the plasmid looks like. Now, what we do is that we have our vector like plasmid, and we have our DNA of interest. Suppose this is our DNA of interest, which we want to amplify. So what? And this is our vector interest. You get my point. Yeah.
Yeah.
So it will be circular. So what you do is that you use restriction enzymes to cut this and cut some ends of this also, and sort of join them together, so you get something like this, if that makes sense.
Yeah, I got you.
Yeah.
So you use
So
restriction
how
enzymes to
does
make
this
the cut
happen?
in it and then attach the stuff.
Yeah. But to understand how this attachment happens, we have to go into how restriction enzymes work. So restriction enzymes recognize a certain sequence and they cut accordingly. So I think NCRT's
Right.
example for restriction enzyme is pretty good. So suppose EcoR1 is everyone's favorite restriction enzyme. So you have vector DNA, you have foreign DNA. You cut with EcoR1, generate sticky ends. Correct. यहाँ से कुछ नहीं है, and you have sticky ends which are unpaired nucleotide. ये इसी भी EcoR1 से कट किया था, तो you have sticky ends with unpaired nucleotides. Sticky ends will join with sticky ends. Are you seeing this?
Correct. Yeah.
AAPT. That is why palindromic sequence was important.
I finally understand.
Yes.
I didn't know one was foreign, one was vector. Right.
So now, how do we connect with our this thing? I think next image दिखाता. So you have your vector DNA, foreign DNA. You cut it with the same restriction enzyme. You also cut the foreign DNA with this thing, and you use. You remember DNA ligase?
Yeah.
Like I told you to remember that, right? Like Okazaki fragments को एक साथ करता था वो. Joining together DNA.
Yeah.
We cut it, but join करके join भी तो करना.
But हाँ वो तो अंदर ज्वाइन हो गया ना अब मेन इसके साथ भी तो ज्वाइन करना
नहीं join उतना आसान नहीं है. Like this looks like अपने आप चिपक गया. मतलब ऐसे लग रहा है कि अपने आप चिपक गया ना. This is what you're saying. नहीं चिपका. वही तो like it's not that easy. Like हाँ है. इतना आसान थोड़ी ना है bro. You're thinking of these are 4 sequence in a million per sequence edge. What is the probability of it coming together? It's very low.
At
You need
least
some
20%.
mechanism to bring it together.
Mhm.
That is what ligase does. DNA ligase brings them together and पास आने पे चिपक जाएंगे. But you need something to bring it together.
Okay. हाँ.
हाँ उधर भी सिमिलर चीज हो रहा था ना मतलब like you had strands which needed joining. So ligase does this ligating, that is joining.
Mm.
So Okazaki fragments वाला same case है यहाँ पे. Now issue happens when like you don't have common like what do you call your vector DNA and your foreign DNA don't have the same cut sites, like they don't have the same restriction enzymes.
Mhm.
So there are 2 things you can do. One is you can cut with like this is where stuff like this this is BAM H1 and BAM BAL 2 wala case that they've given. This was the case that given, right? BAM H1 and BAL H2 wala assignment.
Yeah. This is the one they asked us to form the restriction mapping to.
Yeah. No. Like they wanted to mention like why such thing such 6 mer wala question, like how many times will it happen?
Oh, okay,
Oh, you
right,
didn't do
right, right.
assignment too, bro. You didn't do assignment too.
हाँ,
इसलिए तो मुझे याद नहीं.
assignment
Bro,
2
I did
also.
everything day earlier and then she screwed me up. Bro, I spent so much time on the assignment. Anyway, which one? Like, so basically they'll produce the same second. So its advantages अगर suppose हमारे vector वाले में there is a BAM H1 site and हमारे foreign वाले में BAM H1 नहीं है but BAL H1 है, H2 है. BAL 2 H2 क्या लिखा है? BAL 2 है. So, like I can just cut with BAL 2 and get away with it because they are producing the same sticky.
Oh,
Doesn't matter.
right. I see. Though the sequence is different, but then
Bro, the
the
sticky
sticky
end
end part is
is going
exactly
to
the
be
same.
TATC. So what is going to be the sticky end for this?
Right, it's the same as this. Right. So wherever those what do you call the straight lines are,
that
right?
cuts. BAL 2 is cutting from A. No, it won't, right? GAATTC, right? What is happening with this C? This C was already bonded with the G, bro. So इसमें A will already be bonded. So इसमें A will be bonded with thymine, doesn't matter. But like the sticky end that is produced is same. मतलब आप ऊपर वाला नहीं देखेंगे, हम नीचे वाला देखेंगे ना. That is C, T. We can get away with this. So that is what we actually do. We get away with it. Like that is BAM H1. No, it won't, right? It won't recognize it. The sequence is different. इसमें GATTCC था, इसमें AGATC है.
Well, the thing is, you can use it interchangeably as long as at least one of them can be used on any one of these, either
Oh,
foreign
one
or
second.
vector, right?
1 second.
Then it
I
should be
think I
fine.
understood Stress's issue. पहले हम लोग एक ही सीजर से दोनों को कट करें थे ताकि सेम स्टिकी एंड बन जाए. करेक्ट. एम्फेसिस ऑन द स्टिकी एंड. इसमें भी तो सेम चीज हो रहा है, सेम स्टिकी एंड्स बन रहा है. आई एम जस्ट यूजिंग टू डिफरेंट. ठीक है. नहीं है हमारे, दैट इज द एंटायर थिंग. हाँ, ठीक है. बैम H1 DNA में भी बैम H1 है. ठीक है. Bro, BAM H1 is only one cut, right? There should be another cut also. So it will be one BAM H1 and the other would be eco R1 or something. Getting my point, right? DNA भी दो end से cut होगा.
Wait 1 second. Why do you need two cuts? You need only one cut, right? Because you're
No, you
inserting.
don't. You
You're not
don't.
removing that.
Like you. Okay, I think I should give you with an example.
No, wait, no. I just have one question. So, in your plasmid, whatever you have as your entire circular DNA, and you make a cut, right? Once you make a cut, you don't remove that portion, do you?
You remove that portion.
Oh, okay. You need to remove that portion so that you have two sticky ends from either side to attach another portion.
Yeah.
Okay. Now I get it.
Because if it's only one sticky end, तो DNA का दूसरा end क्यों? Like where would it go? Like it's just a free floating end, right?
Right.
Like if I had
I
only
see.
one sticky end, the other end has to attach somewhere, right? Otherwise, it's not a plasmid. Bro, I'll give normal because abhi, I think I'm getting the feeling that you're not understanding the normal itself. हमारा ये प्लास्मिड है and this is your DNA. Cool. Suppose हाँ इंसर्ट करना है. BAM H1 से कट किया मैंने. ठीक है. This produces sticky end corresponding to BAM H1 here. ठीक है. मैंने Bro, BAM and H1 DNA के अंदर नहीं है ना. BAM H1 is a restriction enzyme which you are adding from outside. Bro, it's just a scissor that you're using to cut it.
No, wait, I didn't get the other side. Like you see, one side has a sticky end. What happens to the other side? Yeah, I assume you should have a sticky end, right?
इस तरफ भी sticky end है. It will also have a sticky end, but we don't
So-
really care about that sticky end.
Yeah, so my point was, if both
Suppose-
sides have sticky ends, why do you have to remove? Why do you need to have two sides?
The reason is that your DNA also need to have two sides of sticky ends, right?
No, DNA has to have two. That is compulsory, right? But the plasmid, if you have this one
vector you
side,
can get away with just one. Vector you can get away
okay, yeah,
with one.
that was my question. That is
Vector you
good.
can get away with one, but DNA you need at least two.
Yeah, DNA you
And
need.
because DNA you request, हाँ, suppose DNA में दोनों तरफ ये DNA था and you were able to break it like this into something like this with just BAM-H1 cuts, then well and good. मतलब तुम्हारा ये वाला part यहाँ attach हो जाएगा, you will essentially get something like this. Correct.
Right.
But better, like in most cases, this doesn't happen. In most cases, like one side is able to get cut with BAM-H1, other side is able to cut with equal. So what you have to do is that यहाँ पर भी equivalent से cut. Yeah. What I'm explaining hasn't come yet, like the other case. So दोनों तरफ cut करेंगे and this gets ligated here.
Okay.
Yes.
Right.
Does it make sense?
Yeah.
अब हमें तो sticky end से फर्क पड़ता है ना. Like suppose I was representing BAM-H1 with this color instead of eco R1. Let's assume that I was doing BAL-H2. Okay, like I'll give the proper example itself. Arihant ने जैसे बोला वैसे हम लोग करेंगे, like in the sense that we have only one cut, we are getting away with one cut. ठीक है. But we have a small issue with दो BAM-H1 के नहीं है, छोड़ो BAM-H1 के है ही नहीं.
Yeah. Got it.
अगर दो नहीं है तो it will cause weird cuts and which might result in sequences that we don't require. We have to be very careful while choosing restriction enzymes. Suppose BAL-H2 के है, it will create sticky ends, green sticky ends. ठीक है. Actually, these are not green, right? Because the sticky ends are actually identical. They're actually red. They are not separate sticky ends. It's
Yeah, I got that. So you just have to pick up any sorts of like restriction enzymes which have similar sticky ends.
Similar property, yeah.
I see.
Like it's not that easy either. Like in most cases क्या होता है, ये भी काम नहीं होता. Like entire thing may not have this working out also. Does it make sense? Like ऐसा कुछ restriction site नहीं होता जो common sticky end दे रहा है. अब क्या करें? So what you will do is that and there are restriction enzymes which give you blunt ends instead of sticky ends. नहीं, direct blunt end देता है, like without any sticky, even if it's parent.
Right.
So these are two major issues, right? What do you do is that you bring our dear old friend ligase again into the picture. Ligase का purpose क्या था? It joins thing, right? Stuff to the end. No, so what you do is that you bring in ligase and you add a molecule called the like adapter molecule.
Oh, right. This has a sticky end, right?
Adapter molecule has the sticky end. You just ligate linker. So adapter molecule is what connects the linker molecule and the like linker molecule is essentially a sticky end. The adapter molecule adapter connects your DNA with the linker. You can't directly attach linker ऐसे. You need an adapter molecule in between. Yeah, and ligase का purpose है to do the attachment. Cool. And that is how you proceed with it. So there are constraints on size. There are concerns on size, though. That is why this is a slightly more complicated technique. You have to do it. It's also in general also more complicated, right? बहुत कुछ करना पड़ता है. So why are we doing it at all? We'll come to that. लाइक इसके भी काफ़ी सारे एडवांटेजेस हैं. We'll come to that. So we have learned how plasmid is working, right? Cool. तुम क्या करोगे? लाइक प्लास्मिड के अंदर इंसर्ट कर लिया. हाँ. Now you will introduce this plasmid into a bacteria culture. In the bacteria culture, and remember this was coding for some antibiotic resistance. You add plasmid plus plasmid. Now you add antibiotics corresponding to whatever resistance you had in this plasmid. So every other bacteria will die except for those who took in the plasmid. Every bacteria which doesn't have this plasmid will die.
Right.
Whatever set you have will all contain your plasmid. Once you have it, you just have to do bacterial culture. Bacteria split होगा, तुम्हारा plasmid भी split हो रहा है. It's the same
Right,
thing.
so
Bacteria.
So now you just culture
You
the
have a
bacteria and
bacteria
let it multiply.
which has a DNA that of your interest. Now you keep on multiplying this. Give it food, put it in a reactor.
You can create a bacterial army.
It gets better. Like
क्या बोला तूने अभी क्या बोला बैक्टीरिया वन क्या
No, I think bacterial army of DNA. Same restriction in them. Right? You know the site where you put it.
so that is all handled by ligase and all. So this is a huge thing. So remember that this had like I'll bring back the image so you will understand क्या हो रहा है. So what you do is that that is also a way, but generally what your thing has is that it would have two like antibiotic resistance together, and तुम्हारे cut sites are within your like resistant sites. So you sort of put combinations in it so that you get only ones which have this cut. For example, अगर मैंने BAM H1 और SAL H1 वाला replace करके अपना like protein डाला था, मान लो, hello, plasmid डाला.
Yeah.
So it will show resistance to only ampicillin and not for tetracycline. So I keep doing this process till I find that my colony is not showing tetracycline resistance. That is how I recognize that it has been transferred. So this is called selectable.
Yeah, but even I didn't get to it.
This is to identify like तूने कहा कि extract out कैसे कर रहे हैं? There are only two ways, right? Either you have plasmid. Oh, you are saying plasmid. अरे you are talking broken वाला part को हटा कैसे रहे? That is what you are asking.
Yeah. It's like whatever
नहीं हटाना
sequence
bro.
change.
तो हटाओ मत. Like bacteria will never take this piece. Bacteria will only take a complete plasmid, right? टूटा फूटा पीस पड़ा, बैक्टीरिया को क्या फर्क पड़ा? Don't take it at all. हाँ वापस चाहिए कैसे कर रहे हो? अच्छा, so वापस चाहिए प्रोसेस restriction enzyme से कट करते हैं. I misunderstood your question completely, bro. I thought इस छोटे पीस को कैसे निकाल रहे हो और समथिंग. So uh, yeah, so if you want to get back this thing, थोड़ा और कॉम्प्लिकेटेड है, लाइक रेस्ट्रिक्शन एंजाइम से कट करोगे, बट पहले यू हैव टू लाइक add proteases which will eat up proteins. Then you have to add other stuff. So basically, you have to eat up the bacteria first, and then extract out and cut.
Okay. It's not like you'll just give out that plasmid as it is. You have to
Yeah.
like kill the bacteria and then take it with force.
Then take it. Yes, with force. So, which is what makes this an entire complicated process. You have to extract out the plasmid again and out. So, but now I come to the bigger advantages of this. Remember, हमें primers चाहिए था PCR के लिए. ठीक है, प्राइमर चाहिए था PCR के लिए. फर्स्ट लेट मी मेक दिस बैक्टीरियल इमेज पहले पूरा. ठीक है, आई डोंट नीड कट्स एंड ऑल. I have an unknown DNA sequence, and I want to get more amount of it. I can't do PCR, right? Because primers नहीं है मेरे पास. It's an unknown sequence, so I'm forced to do cloning. That you can do experimentally, right? Try with multiple restriction sites and so on. Exact चाहिए. Exactly. You need the exact sequence that you want to do. So, so that is one. That is one advantage. And second, यहाँ से sequence करना आसान है. Like if you want to do PCR, once you do this, that's easy. How do you do PCR? You cut it somewhere here, this plasmid. Now it's a straight DNA, like with your thing in between. And you do PCR with, primer, इसका sequence, right? Plasmid का sequence. So just do primers with plasmid का sequence.
Oh, I see.
And then do Sanger or something. इसका sequence तो पता है, black वाले का.
So wait, all this we had to do was just to see if it had antibiotic resistance, right?
We had to do is check whether it has antibiotic resistance to make sure that
once we know that it does, then we can do a PCR to amplify the sequence that we want.
I mean, why would we want to do a PCR? We can just let the bacteria grow also, right?
Yeah, I mean, no, you're saying cut it and then put primers there, right? Okay, wait, that is to identify the sequence you mean. Like, what was the single strand thing you did?
Generally, what is done is that you want to sequence it, right? It's an unknown sequence, correct? So,
no, but what you inserted, you knew, right?
Don't know what you inserted.
Oh, that also you don't know.
Yeah. That's why you're doing
this. Okay, so everything is guessing. Okay.
You wanted an unknown sequence and you want to sequence it. You can't directly do it as PCR, right?
Okay. No, I assume
You want to do a standard sequencing.
Yeah, I assume whatever we are introducing into the vector, we knew. But that can also be unknown.
Like, imagine you discovered a new species. You don't know what its DNA is like. Correct.
But you just want to know whether it has antibiotic capabilities.
No. That doesn't matter. Antibiotic capabilities was within our plasmid, right? It has nothing to do with whatever we are inserting. It's the property of the plasmid that we selected. Like, is यहाँ पर कहीं antibiotic? So antibiotic resistance was something that was in a vector plasmid. Why? So why is it called vector? Because it is the vector for, like mosquito is called the vector for malaria and all, right? It's a carrier for
Right.
like your like gene of interest, the DNA of interest. So what was your question?
Oh no.
It carries it. हाँ, उनके जेनेटिक मटेरियल में ऑलरेडी होता है लाइक टेट्रासिलिन रेसिस्टेंस एंड एम्पिसिलिन. दिस इज नॉट समथिंग वी हैव एडेड, पहले से था. डज इट मेक सेंस अरिहंत? ओ कौन सा मस्कीटो की बात कर रहे हो क्या? Oh no, mosquito is, I was giving you an example. Like vector is used in terms of carrier. That's it. Mosquito is nothing. No, nothing of the sort. No, I was just explaining why it's called vector plasmid.
Okay.
It's called vector plasmid because it's a carrier for our information thing, and it already has tetracycline and ampicillin resistance. ये हमने ऐड नहीं किया, पहले से था प्लास्टिक में. This is why we chose the plasmid also, right? अगर plasmid में कुछ नहीं है, how will we select कि कौन सा survive करेगा, कौन सा नहीं करेगा? Remember, we killed all the bacteria to get back the ones which changed, transformed. The technical term is transformed, so which will contain this plasmid. So these resistances are called like tetracycline and ampicillin resistance are called selectable markers because this is how you select the bacteria. Yeah, so that is clear, right? So it has nothing to do with what we are adding. हमने add ही नहीं किया. This was already there.
Okay.
Now, as Shreyas rightly put it, you can directly say something like you have an unknown DNA, so you add it to a known DNA, and because we know the known DNA, you use that to clone the unknown DNA. That is what you're doing.
Repeat. You have unknown DNA, you add it to known DNA.
You have an unknown DNA which you want to sequence. You can't do
Right.
PCR directly because you would need to know what the sequence is. So like it's a catch-22. You can't know you can't do sequencing without knowing how to sequence. So what do? Instead, you like take a known DNA, insert our unknown DNA there, and then do because you know the DNA, you know this DNA, you can make design primers for it.
Okay. This had nothing to do with a vector and inserting into bacteria, right? This is a different thing
No.
you're explaining.
Yeah.
Okay. Then I got it. I was confused how that was related to inserting into bacteria. Oh. Okay, so then you just break it up and then, or you don't even need to break it up, just add primers to that and figure out what sequence you add.
Yeah, you need to break it up,
Okay,
right?
okay.
Because circular DNA से कुछ कर नहीं सकते तुम. Like.
Wait,
So
why can't you do it circular?
Because the resulting DNA would also be circular. And second thing is that it's a coiled DNA, right? So how will you do PCR with circular DNA?
Oh, right. You need.
You're doing PCR, right? This primer is coming for PCR.
So wait, even if you have circular DNA, the two strands can't separate.
It's harder to separate, right? It can be
harder
that
to separate.
they don't separate at all, also, right? Because they're coiled together.
Yeah, because they're coiled, they might not be able to unwind.
Unwind. Like you might get a ring with ring sort of.
Right. I get what you're saying. Yeah.
And also, it's harder. Like you have to put more heat because they're much more close together and all.
So it's just easier to break.
Easier to cut it. Yeah.
Okay.
Again, Sanger wala, like if it's a circular DNA, how can you describe what is first, what is second?
Right. Okay, yeah, it just makes a lot of sense.
Yeah. So vector cloning and so this is one application. Like unknown DNA, you can sequence it using vector cloning. And vector cloning, say you can also do amplification, like you just let the bacteria grow on and on. But the best part is that, yeah, best part is that this is a DNA within the bacteria, right? Correct. So the bacteria will continue to express that DNA, correct?
Hmm.
It will produce proteins corresponding to it.
Hmm.
ठीक है.
Yeah.
So we are getting an output of proteins. What if that is the protein that we want for some reason? Like
We get protein also.
yeah, you force the bacteria to make proteins that you want.
This is like
You to
using,
make products you want.
bro, this is just straight up using bacteria.
Yeah. So पहले क्या होता था? So पहले क्या होता था कि for example your this thing insulin is the most famous case. Insulin uh, insulin was obtained from the pancreas of either cows or pigs for human consumption because they are closest to us. It was not perfect, but like it was close enough. It didn't create too many allergic reactions and so on. It was not perfect. I mean, but it was like workaround sort of situations. You can't just go around cutting up humans and taking their insulin.
Uh,
But now what happens is that you force bacteria to produce human insulin. Easy. प्लास्मिड के अंदर इंसुलिन का जीन ऐड कर दिया.
and that produces insulin by itself
That is,
because it has
and this
it.
is cultured in a huge tank. Like it's called a bioreactor. उसके अंदर bacteria is provided food, like optimal climate and all, so that it reproduces as fast as possible and creates more and more of protein. So huge bacterial colonies making insulin. And this is not just insulin, like lots of proteins. If you have ever had protein drinks and all, most likely it has come from a bacteria. Whey protein drinks वगैरह, they would have पक्का come from a bacteria.
The bacteria is OP, they're making food.
So like E. coli is used for insulin, like in case you're interested.
Yeah, but you know.
Yes. I mean, no. कितना डेंजरस होता है इकोलाई? पेट खराब हो जाएगा.
पेट खराब हो जाएगा, like डायरिया हो जाएगा, उससे ज़्यादा नहीं होगा.
Yeah. I have a feeling
Interesting.
yes, randomly रात को उठ के ऐसे इधर उधर देख के बोलेगा हाँ हाँ बैक्टीरिया fuck you. वाह. और हम जो खेती करते हैं वो प्लांट्स को भूल ही गए. नहीं, प्लांट्स लाइट बैक्टीरिया.
So like this is how bioreactors will look like. Like they are given air, they are given culture growth, and like thousands of liters of stuff is cultivated at the same time. And it's very cheap, right? One-time expense. तुम्हें एक बार clone करना है, and that's it.
Yeah, they'll keep making the more clothes by
They'll keep
themselves.
making as long as we're giving stuff to it.
Damn.
Yeah, now I can get very philosophical about this and talk about communism, but let's not get there. V and T. No, this thing has not ended yet. Yeah, variable number of tandem repeats. So like this topic is not over yet. Like, there is one small topic which I can see in one line, that is instead of bacteria you can use yeast also. It's the same thing. So the difference is that ट्रांसफर करने का मैकेनिज्म डिफरेंट होता है एंड इट्स अ लार्जर व्हाट डू यू कॉल larger DNA.
Larger DNA strand.
What is the advantage of having larger DNA? You can have larger strands.
More restriction enzymes, let's say.
No. You can have more size inside. That is why. Like,
okay.
You can have longer strands within the yeast wala thing. So,
okay, so a limitation of this would be that you can't do very large strands.
Yeah. Bacteria.
Yeah, you can't clone very large.
But bacteria replication is faster, so like, it's speed over amount.
I see. So I've so the first
like,
time you study it like
I'm flattered. Yeah, I'm flattered very much. But this is also interesting, right? Like,
yeah. I mean, I see real life application within this also. It's not just like I see theory and I'm mind blown.
if it's bacteria, we call it bac, and if it's yeast, then it's called yak. Because I just wanted to finish with this topic and then we can talk. So bac stands for bacterial artificial chromosome, and same thing, but yeast हो जाएगा यहाँ पे. Yeah, that's it.
Oh wait, you're done.
No, VNTR wala. So VNTR is the last thing that we have to cover. That is basically how do you, given a genetic material, how do you identify individual, where he's from, blah 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 blah. This is
the whale
which
and blubber
individual
thing.
Killian whale blubber. So like Shreyas mentioned that 90% of our DNA is same as that of banana, right? I'll say that 99.9999. Okay, I have to type. I have to do till 10 places. So up to 10%, 10 places of decimal, your DNA is same as every single human. Everything is same. Nothing, not even one base will be different. So like most of your genetic material is the same. But we see so many differences. It doesn't make sense. So what? Yeah, that's a fair enough
kind of
way
a
of
view.
thinking. Yeah. It's a nice way of thinking. GG. So how this is basically coded is that तुम्हारे chromosomes like के end में there are these regions towards just the ends are called satellite
chromosome की
DNA. So satellite DNA can like look like it according to size. It can be satellite, microsatellite, and so on and so on. Okay, like size-based differentiation. Now, how the satellite looks like is that it has lots of repeating patterns. Same pattern repeating over and over again. For example, TA repeating over and over again for millions of base pairs.
Okay.
Like similarly AAA. So TATA is a special case called Tata box, and you have something like GTAA, GTAC. These are all common like repeats. You also have stuff like uh, A, like just repeating on and on forever. Like,
Mhm.
so these patterns are called tandems. And VNTR, I don't even need to explain what VNTR is. Once you hear it, you'll understand. Variable number of tandem repeats.
Yeah, I think you either mean the number
Bro,
of base
it
pairs
is a
in each
variable
tandem.
like across from, yeah. So number of occurrence of each tandem repeat will be different from one individual to the other because this is the thing that is different from one person to other. So with VNTR you can pinpoint कौन सा individual है क्या individual है.
Or you mean the tandem sequences are the same, but the number of such uh, like
Yeah, these
repeats
sequences are
is
same,
different.
but like this thing can be like 100 times or 1,000 times and so on, so on, so on, right? Or
Mm.
it can run into millions. In some people, it
So
might not
then,
even be present.
okay, the number of times it exists will show certain
Number of
features
times
over
each
the
of the
other.
tandems are repeating is VNTR, like variable number of tandem repeats. Same. Yeah.
So if
So these numbers are actually different.
huh?
Yes.
And
Yeah.
can
So
I?
wait, so
Does that mean can I fix my Valorant addiction?
I mean, it just means that it's closer to monkey than you realize.
so when you say it can repeat 100 times to a million times, do you mean it's one continuous sequence of that particular
Yeah, yeah, yeah.
subsequence
It's a
occurring over?
continuous sequence. So it's like
Okay.
Tata box अगर repeat हो रहा है तो it will be like this sequence all over again and again and again.
Okay, okay.
It's like uh,
So you mean the length of that sequence, basically. That's it.
number of times this.
Yeah, that is directly proportional to length. Great. So that's what
Yeah,
matters.
yeah, yeah.
I
So
see.
this, bro, the name itself gives it away, right? Variable number
Yeah.
of tandem repeats. Tandems
Hmm.
are these. There's a variable number of like tandem repetitions. This is how you
Nice.
pinpoint on individuals, and and this is how you do one second like DNA fingerprinting वगैरह इससे होता है. Like you compare tandems of like parents or like if you're doing genetic testing or something, you do parents का thing and you compare with offspring का thing, and then you'll find oh तो इसकी का बच्चा है या नहीं है बच्चा or something. Or if you have multiple people who you are doubting, you will just compare the tandem repeats after doing a gel electrophoresis sort of thing.
So is this how you
Yeah,
check if someone is a child of a parent or something? That
Yes.
DNA
yeah,
test is
yes,
tandem repeats.
yes. So tandem को sequence करता है. Bro, they like sequence the tandems and 100% similarity नहीं check कर रहे. They are looking at which shows sort of
Closest.
similarity. That's a
Huh.
threshold.
Interesting.
And the probability of two people having the exact same tandem is literally zero, right? Because of the sheer number that we have.
Hmm. Maybe if one particular tandem, it might happen, but definitely not all.
Not all the tandems.
Right.
It's
Imagine,
not literally zero, it's like approximated to zero, sort of.
Yeah, yeah.
Yeah.
imagine if someone had exactly same thing as you, like lived
They would be you.
maybe 100 years ago.
Nice.
It's theoretically possible, but the probability is pretty low.
Hmm. Yeah, if you take
हाँ. Hmm. No, it will happen everywhere. So this is not the this is the variation among humans, bro. Like, yeah. So evolution कैसे होता है? Evolution is actually due to when we learned about replication process. We were talking about how polymerase is working and all, right? Polymerase also makes mistake. It's a very reliable molecule, but it also makes mistakes. वो गलती से T के जगह A डाल देगा या ऐसा कुछ हो सकता है. ठीक है, एक जगह पे वो चेंज कर दे गलती से, बट बिकॉज एक जगह पे हो गया, रिमेंबर आवर रीडिंग फ्रेम.
Yeah, that
If you
three
insert
three
something by
that will bring.
mistake,
So your
entire reading
entire
frame will
amino
go.
acid. Yeah, that protein will change.
Some new protein will be produced, or some like
Yeah.
protein will stop being produced.
Yeah, one protein that you required is no longer there.
So that's how small changes are built. If there is a large number of changes, so for example, if you are exposed to UV radiation or some other like thing, it basically alters a DNA, causes errors, but like that is too much of error, right? That's why like people will be born with mutations, like very high large step mutations. In fact, most of them will die. Like the children that they will have will end up dying. That is because the mutation was too large. Evolution will only happen if the mutations are very small at each step. Because if you have a large mutation, then some essential function is being deleted without some complementary function being developed. Yes.
Or you got infinitely retarded.
Like I'll give you an example. So have you heard of sickle cell anemia?
Yeah.
So sickle cell anemia,
Exactly
I think ITP में हमने किया था, like it like sickle shape में होता है under oxygen pressure.
And it cannot,
Like if you have
yeah,
less oxygen,
cannot
then
carry oxygen
your
properly.
RBC loses shape, and it cannot. You end up choking if you have less amount of oxygen in your blood. So it's a very sad disease. So it is caused due to one insertion, just one insertion in the 12th chromosome.
So I mean, the fact that we are proper and healthy itself means that it's a miracle in copying. Yeah.
Thing is to remember is that हमारे में changes बहुत slow होता है. It's that is what basically everything is building upon, right? Everything is very slow. Like changes that happen is very slow. If large changes happen, we'll die. If large changes don't happen, we'll live to see another day, like another generation actually.
So,
So
you know, there was a way to
what?
kill. Huh? No, continue.
So if there was a way to, no, I was saying that even from the bacterial colony to शुरू में हमने RNA to DNA transition, RNA to RNA transition, everything is a matter of survival. You choose in a way so that your genetic material survives, lives to see another day. Correct.
Okay. Yeah.
And that is basically what life is all about.
Yeah, see, if you want to kill bacteria or you want to prevent bacterial growth, you do have antibiotics which which you can introduce to them, and then that will block some kind of production of some enzymes, or kind of kill it by eating salmon, some kind of procedure, some pathways there. But if you want to kill a virus, right? Virus basically just has RNA in it. So you have to introduce some sort of mutation that will stop it from using or coding for the current protein that it needs for its survival, and then kill it. That is just one possibility, right? For killing
Yes,
virus.
that is a possibility. Good that you mentioned it, because recently there has been lots of interest around something known as gyrons. Like maybe Alapan also has read about this. Have you? It's gyrons, Karan, gyrus. Dada, have you read about it? Gyrus is nothing complicated. It's just giant virus. These are viruses which are
Interesting.
as big as bacterial cells,
Oh,
which is like 1,000 times bigger than what we expect from general viruses. What makes viruses even more interesting is that they are attacked by viruses too.
interesting. Huh.
So you have virophages which attack viruses. So this blurs the line between what is dead and living even more.
Hmm.
So how like viruses viruses also cause diseases in humans, and also like like how do virophages basically work? They do exactly what you said. Like they hijack its protein and they kill it. And now treatment methods are being formed where like you introduce virophages which will have some sort of thing that will kill the virus. In fact, lots of primitive bacteria and all on which the virus preys also has like that sort of bacteria, virophage or DNA in it, so that if the virus comes inside, like it will end up killing itself in a way.
Oh, I
मतलब the
see. Huh.
one organism will die. Like suppose it goes into this bacteria. बैक्टीरिया के अंदर that virophage का थोड़ा डीएनए का पीस होगा. ठीक है. This virus will come inside, it will hijack its machinery, but while replicating, it won't be able to replicate more. This cell will die. This virus won't be able to attack more cells.
Oh,
Does it make
okay.
sense?
Okay. Huh. It kills it and takes it down with it.
Yeah. So the bacteria is sort of saving its entire species in a way, taking the hit for the species.
Bacteria, smart, bro.
Bacteria has to do that, right? Like it can't sense much of the world. It doesn't see the big picture. Maybe we don't see the big picture either.
Yeah. Yeah, it's like it does everything as a group. It does not think anything selfishly. Yeah. Damn.
You know that there are no true cures for viruses. Like
Yeah.
you always treat symptoms or something that promotes your body's attack on the virus. You can't really do anything for the virus.
Damn. That was nice.
Yeah. I mean, that's it. So, one second, I forgot to say one more thing, and then so blubber whale वाले example में she used a specific example of mitochondrial DNA. And so mitochondrial DNA का case ये है कि like let's learn reproduction again. You have egg, you have sperm. That's how life is made, right? And this like fertilized egg splits into smaller clumps. Like fertilized egg is called what?
A zygote.
It's called zygote. Yes. GG, nice time.
Yeah, nice.
Yeah.
The reproduction is the gem.
Yeah. So fertilized egg is called zygote. Zygote keeps on making clumps, and it makes what is known as the fetus. Correct. So basically, essentially, तुम्हारा like how it works is that your sperm embeds itself inside the egg, correct, and then it releases your genetic material, and the sperm cell is sort of it dies, like it has no other role once it releases. ठीक है, genetic material is released within the egg, fertilized egg. Now your mother's cell is going to replicate, divide and all, right? So all of your mitochondria that you're going to receive is going to be from your mother's end. Remember, mitochondria had its own DNA and all.
DNA.
It's
Right.
passed from one cell to the other, but not from parents, right? Like मेरा DNA has nothing to do with mitochondria का DNA.
Mm-hmm.
So the baby receives the mitochondria of only the mother and not of the father. So there is a direct link between the child and his mother, maternal side actually.
Right.
Now you can use this to analyze, like after taking enough samples, you can use this to analyze whether it's a female-centric society. Are there more females than males? What is the relationship between females, and so on and so on.
Wait, how can you analyze female society?
Huh. So, like you collect mitochondrial DNA data from multiple people, and you look at what kind of like similarities are there.
Oh,
If
okay. You mean you don't know the population or the population distribution, but if many of the
yeah,
mitochondria are similar, that means if everyone has the same mother, so
yes,
the population of moms are less.
yes. Ka genetic material. What advantages does that give? Because we have a direct link as that being of the mother. Otherwise हमें doubt है कि ये father से आया कि mother से आया.
Mhm. That can give you like the distribution of mothers in the
Yes,
population, right?
it will give you distribution of mothers. It will give you like how many females are there in the population at all. Like
Mhm.
marine study के अंदर like एक जगह से तुम multiple लोगों के लोगे and eventually you see that हाँ there are 4 distinguished patterns. So there are 4 unrelated females there.
Ah. Oh, that's crazy. And she actually teaches this stuff in class. That is even more mind blowing.
Hmm. Hmm. हाँ. Hmm.
No,
नहीं
no,
ना,
but
that is the beauty of
mitochondria does
mitochondria.
has nothing to do with your cell
Mitochondria has its own
DNA.
DNA.
So it will
Mitochondria has
reproduce
its own
and
DNA. Ah, mitochondria is like a cell of its own. That's why we. Mitochondria.
uh,
Uh, okay, I'll tell you one more thing. It's not actually very practically useful. इसलिए like ma'am hasn't mentioned it either. That is plastid. Have you heard of plastid?
yeah, it's present in plants.
Oh,
Leucoplasm,
plast.
some I remember some stuff.
Plastids and mitochondria have one thing in common. They both produce energy, which also tells something very fundamental about life. We'll come to that later. So plastid is basically what does photosynthesis.
Mm-hmm. Right.
It is, and it also has its own DNA, ribosome,
Chloroplast.
and then
Huh.
everything. Yeah, chloroplast. No, kind of. We sort of ate them, and we realized that they are good energy sources. They can create energy out of nothing. So, so we ended up keeping them. See, life seems so complicated, right? When you think of all of this,
It is, yeah.
but somehow it worked out. How crazy it is that everything seemed to work out, and exactly, man. How crazy is that? Like some, bro, forget everything. This world was built from scratch, man. Think of it. Everything that happened, किसी ने किया नहीं, अपने आप हो गया. Chemistry भी अपने आप हो गया.
Yeah, just that we are learning these things, but they were always there.
They're always there. Biology was always there. अभी तक discover नहीं किया. Like I don't know. I enjoy biology a lot because I find that something blows my mind every day. Like something that I didn't connect before suddenly clicks. Hey, bro, I feel the
Uh,
same thing, so I know where that will go.
Bro, it's technically not even close to late because you're still in the UG phase. You can even him. Hmm. Bro, same. Whatever
Bro,
Rishi taught us
if you
in
want to
ITB
learn
is
bio,
first.
I'll give you the best source ever. Like, matlab, it will cover 11, 12 का bio NCERT mostly. That is NCERT. And NCERT is more than enough, as you have realized.
Yeah.
So that is Khan Academy's bio playlist. Pretty good. It's it's nice.
But yeah, I
Yeah.
think I went through a lot of Khan Academy. And this transcription translation I read through Khan Academy in ITB.
Like what I'm saying is that don't restrict yourself to that, but like go through it. Like that will clear up lots of concepts.
So yeah, for sure, because
I'll give you an example bro. Like इंडिया में हम लोग 12th के बाद डायरेक्ट एमबीबीएस चलते हैं करेक्ट इन यूएस इट्स अ मिड career choice like when you're 40 or
Mhm.
45 you decide that you want to become a doctor
I
yeah
think
or
I've seen
like
this also.
hmm,
Like even if you want to be a
it's like
lawyer or something, you can do engineering and then do law, right?
yeah exactly so very few people actually just directly become even law may be a side like you can't do it without bachelor's
Yeah.
law business school everything requires bachelor's before joining
And it just makes
which is a
sense.
good system I feel.
Yeah, I mean, by
Yeah.
then you're sure of what you want to do also. Yeah, it's kind of early. No, but yeah, I mean, as soon as after ITB and just in general about how drugs and vaccine, all that development is done, I was like, yeah, fine. Drug development and drug discovery. It is.
So the very reason why I didn't take drug discovery was that um, it's chemistry more than bio. So yeah,
Yeah. So yeah, see, like,
I would be more interested in something directly linked to bio, like say I don't know, some computational model of biology. Or,
yeah.
but Nita is not doing my stuff right.
Well,
So
that is
Nita
a
is
good
practically
thing
useless as
also.
far as.
I don't know
Uh,
what she teaches in class. Like, see, if I have to learn by myself, I know I can do chemistry, okay? Like, I know I can understand it by myself and do it in depth. But it's not that I can't do bio, but I need like a perfect teacher like Rishi to teach bio. If I have to learn the stuff by myself, I would die.
Arey bro, I'm flattered. Don't,
Yeah, that's the
bro.
thing.
I'm just flattered.
No, no, it's not like we mean to make it sound flattering, but I'm not even kidding. If I had to do this all by myself, I am almost certain I wouldn't even have understood half the things you taught us today, even if I read it for like 2 days straight. Yeah. But yeah, once
Yeah, it took time, but we covered.
huh?
Like the thing is that the math, when I'm teaching you, I know you people very well, right? I know what during my own explorations I found something interesting, right? So I want to share what I found interesting. Yeah.
so yeah, further the
So
connect is
that is
also
the
there.
main difference. That is.
I mean, you know how you liked it and what method of teaching would be right because that's what worked for you, and most probably that's what will work for us.